Dear guests, I welcome you to this annual May Fulbright Forum, the first of two that we're hosting this week. This is an event that we always look forward to because it's always so interesting and I always learn a lot. Even though a number of scholars have already presented, this year we have so many presentations in May that we decided to divide it into two forums. One for our fellows and one for the scholars. So today we get to hear from all of our US Fulbright Fellows, students who have spent almost 9 months in Iceland, either in a university study program or doing research. So I'd like to give a warm welcome to our 8 student fellows who are featured here today. Let me note also that the event is recorded and those who are joining us virtually can type their questions into Zoom. Through Fulbright Iceland, we offer our outstanding grantees an opportunity to grow professionally and as individuals. Being a Fulbrighter is a privilege. And our grantees really use this opportunity to the fullest. The grant period is a learning experience, but also an opportunity to share their knowledge and insights and to engage locally, to share the best that the US has to offer, and to immerse themselves in the culture of Iceland. They really are ambassadors who work to increase mutual understanding between the US and Iceland. They build bridges, collaborations, and lasting friendships. Alas, all good things must come to an end, and our fellows will be finishing their grant period soon. So today, they're going to tell us a bit about their experiences in Iceland. As I noted, we have 8 students presenting today. This is an impressive group from a wide variety of fields. They're all engaged in interesting and impactful projects. It's always a treat to hear about the diverse projects of any given Fulbright year and experience the perspectives of our US grantees. So without further ado, I'm going to get us started. We have 4 speakers in the first segment before the break. They're, again, involved in very diverse projects. It can be hard to be the first one up to bat, but I'm sure that Mia is up for the challenge. She comes to us from the University of Minnesota, and she's here studying in the master's program in geology at the University of Iceland with an emphasis on renewable energy. But I know she's also done a lot to immerse herself locally in the culture. So let's hear from her directly, Mia.
Hi, good afternoon, everybody. My name is Mia, and today I'll be discussing my Fulbright project, Climate Control from the Ground Up, Geochemistry of Geothermal Systems in Iceland. So how did I end up here in Iceland? Well, I'm a master's student at Háskóli Íslands, or the University of Iceland, studying geology with a focus in renewable energy and geochemistry. And the reason I chose this is because I'm very passionate about geothermal energy, and Iceland has a lot to teach the US in terms of geothermal energy. So if we take electricity production, for example, Iceland produces around 30% of its electricity from geothermal energy. But let's look at the US. We produce only 0.004% of electricity from geothermal energy, but our reserves are a lot larger than that. So I want to play a small role in this greater mission to decarbonize our energy sources and work towards increasing geothermal energy in the United States. But this isn't the only reason I'm here in Iceland. Of course, I want to experience live geological processes, and what better place to experience eruptions and earthquakes, and also learn Icelandic and the local culture. So this year I've taken a full load of coursework split into 2 semesters. In fall 2024, I took Renewable Energy Introduction, where we did a broad overview of different renewable energy sources, and we visited a variety of different power plants. In Geothermal Reservoir Physics and Engineering, we looked at how geothermal resources are estimated and utilized in a computational manner. In groundwater hydrology, we looked at the fundamentals of groundwater flow on the subsurface. Geophysical exploration was a lot of field work, so we were able to go out and use a lot of different equipment to look at the earth without being very invasive. And I took basic Icelandic 1. And then in spring 2025, I took geochemical analysis. So we use a lot of different instruments to measure different properties of solids, liquids, and gases. In climate change, past, present, and future, we did a lot of literature review and discussion to understand how climate has evolved in the past and what this means for the future. And this put a lot of my work in context to understand why exactly we need to move towards renewables. In metamorphism, hydrothermal alteration, and weathering, we looked at lots of different rocks, especially those that have been altered by geothermal processes. An interdisciplinary group project within renewable energy was a collaboration between a local company and the university working at the Afforestation power plant up north. In seminar on current geological events, we discussed every week about current geological happenings, and I continued with basic Icelandic 2. And a lot of people ask me, what does geology coursework look like? So this is just a small snapshot from the geothermal reservoir physics course, and we're estimating the resource capacity using a variety of different techniques. So mathematical techniques, computational, and geophysical. So it's a lot of different integrations of things, both numbers and visualizations. But of course, I also look at a lot of rocks, and this is just a close-up of a geothermal alteration mineral that we looked at in my geochemical analysis course. And this is another way that people look at rocks from the metamorphism class, where we take very thin slices of rocks and put them under a microscope. And these different colors and textures can tell you a lot about what happened to the rock in its geologic history. And this is very commonly used in geothermal exploration. And then finally, this is me taking a final exam in front of the glacier Sólheimajökull, which is very, very Icelandic and very fun. And in addition to all of my coursework, I'm also preparing for my thesis work next year. I'm working with my supervisor, Dr. Samuel Scott at the University of Iceland, as well as collaborating with Orkuveitan or Reykjavik Energy. And I'm looking at low temperature geothermal fields, which are reservoirs with a temperature less than 150 degrees Celsius at a depth of 1 kilometer. And these resources are so important because they're utilized for district heating here in Iceland. So they help us get our heat from the radiators and the swimming pools and greenhouses. And to be more specific, I'm looking at the Ivie geothermal field, and it's experiencing 2 main issues. So it's cooling on the degree of 10 Celsius per decade, and there's also oxygen infiltration, which can cause corrosion of the pipes. So to better understand why this is happening, I'm doing interdisciplinary work. In this past semester, I've tagged along with Orkuveitan's sampling team to sample the fluids in the field. So I go out into different well houses like this and collect the fluid and then bring it back to the university to measure different properties of this geothermal fluid. And then in the future, I plan to do geochemical modeling of mixing dynamics, and this will help me to better constrain the mechanisms and timescales of the cooling and chemical changes in low temperature fields, not just in Iceland, but also in the United States and around the world. So I ultimately hope to improve the sustainability of utilizing these geothermal resources. And just kind of to orient you, this is the study area, and it's very close to downtown Reykjavik. So it's a very relevant area for all of us. But in addition to all of my geology, þetta ár hef ég verið að læra íslensku í íslensku grunnur 1 og 2 og mér finnst mjög skemmtilegt að læra íslensku en hún erfið. Og markmið mitt mig langar að æfa íslensku meira með vinum mínum. Ég vonast til að geta talað íslensku við samstarfsfólkið mitt. Ég vil horfa á fréttirnar og sjónvarpið á íslensku. auðvitað að geta hlærað Íslendinga. So basically, I want to eavesdrop. And going back to some geology, I did a lot of fieldwork, especially during the fall. And this is from the geophysical exploration course. So here I am wearing a proton magnetometer on my back, and we walked back and forth throughout this lava field to identify where a lava tunnel was. And essentially where the magnetization was anomalously low, that's where we hypothesized the lava tunnel to be. And we corroborated this data with gravity data. So we took measurements at specific points to identify where gravity is high and low. And we were able to make a pretty good map from this. But in addition to this, we also did some seismic refraction measurements. So this is me hitting a metal plate with a sledgehammer. And this generates small seismic waves, which can then be processed and used to determine the different layers underneath our feet. And another similar method is resistivity measurements. So we inject a current into the subsurface, and this helps to identify the different layers and where the water table is in the ground. So all of these are very non-invasive, and they help us to better understand what we're standing on top of and how it can be utilized, especially in engineering purposes and in Iceland, where the ground is constantly changing. But this is not the only thing I did. I also went out sampling, and in these well houses for collecting samples for my thesis, you really have to protect yourself, both your hearing and your body. The well houses are very loud, especially when they're producing this fluid, and also the fluid is near boiling. So you really have to make sure that you don't burn yourself. So it's nice when it's especially cold out to be wearing all of this. And of course, I celebrated lots of holidays here. This is the 4th of July, not 4th of July. This is New Year's here in Iceland, which resembles the 4th of July. Very exciting. And I'm also a very proud member of the Jewish community here, so I participated in a lot of events. So this is the menorah lighting for Hanukkah, and also some more somber events like the Holocaust Memorial. And this is me lighting candles in front of the Prime Minister, Kristrún Frostadóttir. So that was a very humbling and very honored to do so. I celebrated with my friends and with the Christmas tree lighting downtown, which is nice when it's very dark out. And of course, bóludagur, where you eat lots of round food in preparation for Lent and Easter. So these are absolutely delicious cream puffs that everyone eats the whole week. And then when it's the midwinter, there's a festival called Þorrablót, where people eat very traditional Icelandic food. And you can go to the store and see things like sheephead, which is very interesting. But the most important holiday of them all is Pizza Tuesday. So this is how I began to embrace Icelandic culture, of course. But not only that, I've also become a huge fan of the bakeries and cafes here. And that's honestly where I get a lot of my work done, including this presentation. And I've also taken up knitting. So this is me knitting in a bar, which I would say is a pretty Icelandic experience. And I hope to get better, but we're working towards a sweater. And of course, I love cats now. For those of you who knew me before Fulbright, I was a very staunch dog person. But being here has made me an absolute cat lover as well. So I've just enjoyed petting my friends' cats and street cats. And of course, attending the swimming pools, no matter what the weather is. I think the colder, the better, actually. But this is Vesturbæjarlaug, which is a pool I often go to because it's close to home, and it's nice to just not have your phone and disconnect from everything, and just have a nice relaxing time. And it's not a time in Iceland without experiencing the incredible forces of nature. This is the eruption in November, which I was fortunate enough to see. This was just about an hour after the fissure actually opened, and you could see these really tall fire fountains. So it's just something that, I would never see back home in Indiana or Minnesota. But I've been able to see 3 eruptions while here. And of course, the aurora as well. It's just, when it's dark and it's 4 hours of daylight, this is definitely a reward, especially it's cold and you just want to stay inside, but this motivates you to get out. So just seeing these dancing lights in front of you is something that, unfortunately, a presentation will never capture. But not all the phenomena are positive. Of course, you get this crazy weather. We had a red weather alert and everyone had to stay inside, and it doesn't capture it very well, but it's hurricane force winds, so you really have to, stay inside so you don't blow away. And of course, a common question I get is earthquakes. So this is an earthquake map just from a few days ago, and you can see the epicenter and the time of these earthquakes. And they are something that become a more normal part of life. And especially preceding an eruption, you can often feel a lot of earthquakes. But even when there is no eruption, sometimes they do occur. So it's something that people often monitor, especially in the geology world. So I think it's become a pretty big part of my life, just checking this daily. And also just taking nice road trips on the weekend and seeing some beautiful nature, because what's Iceland without nature? So this is Kjötgjöfell, one of the iconic mountains of Iceland, and Fruafoss, among many, many other pictures that I could show you. But fortunately, my time in Iceland is not over, and I have a lot planned, and I'm very excited for it. Let's see. Yeah, this summer I will be joining the resource monitoring and sampling team with Landsvirkjun of the Deglaciation area. So I will be living at the power plant 5 days a week, and then in Akureyri the other 2. So if any of you are up north, absolutely please come and visit me. I've never lived at a power plant before, but I'm very excited to do so. And I'm also very honored to receive the Leifur Eiríksson Foundation Fellowship for the 2025-26 academic year. So this will support the remainder of my master's studies. And also I aim to complete my thesis work and graduate in the spring of 2026. So a lot is slated, but the least I could say is takk kærlega fyrir. Thank you so much to everyone that has been here, especially those of you in the room and on Zoom. And least not to Fulbright for providing me this incredible opportunity to be here in Iceland and to pursue my graduate education here, and to the University of Iceland for providing me with amazing mentors, both professors and classmates, and the University of Minnesota for allowing me to begin my journey here in Iceland and to continue it with Fulbright. To all of those back in the US, my loved ones, for talking with me no matter what time it is, and for coming to visit me. To all of my friends here in Iceland and the Jewish community, and of course to Iceland itself, thank you for letting me call this place my second home. And with that, are there any questions?
Have time for a couple of questions. If anybody, I can start by asking, so you showed us how little geothermal is being used in the United States. Do you have areas in mind particularly that you're thinking would be good places to explore this more on the US side?
Absolutely. I mean, the West Coast is where you have these main geothermal hotspots, especially Nevada. I think around 10% of their electricity is actually geothermal. So they're really working towards it. But I think the best way we can increase geothermal is by targeting these areas that aren't so much considered to be geothermal hotspots, and using more low temperature technology. And that's in a way why I chose to focus on low temperature geothermal here, because that's more applicable to a wide variety of geologic areas that are actively rifting like Iceland. So I think we need to start taking a look at places, especially like the Midwest, where I'm from, where you don't really consider it to be geologically active, but there is possibility for geothermal in many places that we don't consider. And I think just looking at areas outside of the West Coast is critical.
Yeah. You said 30% of Iceland's energy comes from geothermal. What's the remainder?
Most is hydropower.
Okay.
Yeah.
Okay, well, I want to say that it has been a great pleasure having you here as a Fulbright student. Hello, and here's a little Fulbright swag, a Fulbright pin, a little gift. And we wish you the best of luck as you continue your journey.
Yeah. Thank you. Takk fyrir.
Well, that certainly was a great start to our afternoon. And next we're going to hear from Bridget, who applied to Fulbright Iceland through Amherst College in Massachusetts. She's been working on a research project on mitochondrial DNA damage hosted by the University of Iceland and also working with the National Hospital, Landspítalinn. Bridget has been quite busy in the lab, but has also taken time for other activities like dancing and knitting. But let's hear what Bridget has to say about her year in Iceland and her studies.
Hello, everyone. Yes, great introduction. I have spent my time here, a lot of it in a lab, but I've been studying mitochondrial DNA damage using this technique called the Northern Lights assay, which was named as an ode to Iceland. And for my title page, I'm very happy to say that this is a picture that I myself took of the Northern Lights, something that I could not have said 9 months ago because I'd never seen them before coming here, and was one of my main reasons for initially looking at Iceland. And so even this in itself is very exciting for me. But I'm gonna jump right into discussing my project. And so for a basic overview, the mitochondria is what? The powerhouse of the cell. And a little more specifically, it's the organelle within our cells that's responsible for cellular respiration. And what that means is it's responsible for taking glucose and sugars and converting it into energy for our cells and for our body. And specifically, I was studying mitochondrial DNA. And what some people might not know is that mitochondria actually have their own DNA that is separate from what we think of as our genomic DNA, the DNA that makes up a human that's found in the nucleus. So there's actually it's about 16,500 base pair long genome that's circular as opposed to our nuclear DNA, which is linear. And this DNA is found in multiple copies per mitochondria, and there are multiple mitochondria per cell typically. In comparison to, typically you only have 2 copies of each chromosome in the nucleus. And when we're talking about mitochondrial DNA, we often also end up talking about platelets. And why is this? It's because platelets have mitochondria, but they do not have nuclei, and therefore the primary source of DNA in platelets is mitochondria. So if you're trying to find mitochondria, platelets are a great source for you to look. However, even though you're like, oh, platelets, you can easily get mitochondrial DNA from that, it shouldn't be that hard to then isolate that DNA and take it right out of those cells. However, it's not as easy as one would hope or it might seem. And there are a few reasons for this. First off, most of the existing protocols that isolate mitochondrial DNA are not compatible with the Northern Lights assay, which is the analysis technique that I was using in my project, and I'll talk a little bit about later. And my lab has been working on this for a while, trying to study mitochondrial DNA, and some past attempts have failed because even though working with platelets, they activate easily in vitro. And what this means is once you take platelets out of the body, they like to activate when you put them in a sort of plastic tube, or even just like keep them in other blood, the platelets activate. And part of this activation is the platelets release their mitochondria. And so when the platelets release their mitochondria, if you then try and isolate the platelets, you then therefore can't get all the mitochondria you were hoping to get when you initially started. And taking steps to prevent platelet activation is extremely labor intensive and is actually very challenging and can also be very time intensive too. And so what our goals of the project was to first develop a protocol to isolate mitochondria from blood in as simple and easy a manner as possible. And this is to keep open possible higher throughput studies in the future to maintain lower costs and lower time and labor requirements for that. And then once we did that, use Northern Lights assay to assess the damage. But starting off with the first goal, this is what I spent most of my time doing, is method development to create a simple way of starting with blood and ending up with mitochondrial DNA. And so how I went about that was first getting platelets from blood. And so we started by, you have a tube of just whole blood that we start with usually about 9.5 milliliter tubes, and you spin it in a centrifuge. And as a result of spinning, it separates the blood to its component parts. And the top part of this is what's referred to as platelet-rich plasma. And so this is the part of the blood that we want. And we don't want anything in the rest of the blood, because that's red blood cells, which aren't helpful for us, and white blood cells, which can be an extreme source of genomic DNA and therefore contaminate our mitochondrial DNA. And so we would remove that top fraction of the platelet-rich plasma. And then this is a step that not everyone tends to do when working and trying to isolate plasma, is we spun that again to really make sure we got rid of all those white blood cells, so that you completely limited the amount of genomic DNA in the final sample of platelet-rich plasma. And part of the reason you need to do this is the amount of genomic DNA in a cell compared to the amount of mitochondrial DNA. The ratio is thousands and thousands of times more. And so even 1 or 2 cells of genomic DNA can outweigh a lot of work connecting mitochondrial DNA, which is obviously not what we want. And so from there, what we did was isolate the mitochondria. And the key part of the protocol, and sort of our aha moment, ended up being this first step of, as opposed to worrying about, oh, let me try and isolate just platelets on their own, let's isolate platelets and mitochondria together. And why we did this is because this allowed us to not actually care about if the platelets were activating and spitting out their mitochondria, because by combining maybe a separating of platelets or separating of extracellular mitochondria as one, we spun it all together down at a high enough speed that would put both the platelets and the mitochondria into the pellet. And this is what finally gave us enough DNA to work with compared to prior attempts by the lab. And then from there, just washing the platelet and mitochondria that we had, we treated it with this enzyme that removes any extracellular DNA, so that removes any DNA that's just floating around in our plasma or potentially stuck to the outside of platelets or mitochondria, and then proceeded with DNA isolation with this kit that is just a basic DNA isolation kit. Again, maintaining simplicity was a goal of the project. And so what we were able to accomplish is, here's a gel showing some of the DNA that I had isolated. Each sample you have on the left is DNA that has been digested with what is called a restriction enzyme that cuts the DNA at a specific sequence. And so for mitochondrial DNA, since it's this known sequence of only about 16,500 base pairs, we know the exact bands we expect and the lengths of those bands. And so that's what you can see here, is the different bands are exactly what we would expect when you look at the sequence. And then next to it is DNA that has not been treated with this enzyme. And the 2 samples on the left are what I isolated with this new protocol. And on the right is DNA that's just isolated from whole blood as is. And so when you treat that DNA with this restriction enzyme, you get what we refer to as the smear, because genomic DNA is such a long sequence, there's so much of it, you don't have any distinct banding patterns. And so our goal, this was an easy way for us to kind of tell the relative purity of mitochondrial DNA versus genomic DNA. And so in being able to see this banding pattern showed us when we finally were able to achieve the purity that we hoped for. And so in successfully doing that, we were able to develop this protocol as we had hoped. And so next step was using the Northern Lights assay to assess damage. So big question of the day, what is the Northern Lights assay, or NLA? And so the Northern Lights assay is a 2-dimensional gel electrophoresis-based assay. And for those who don't know, what gel electrophoresis is actually this image that I just showed you is an example of 1-dimensional gel electrophoresis. That is a technique used to separate DNA based off of the size of the DNA fragments in your sample, where smaller fragments are able to travel farther down into the gel. So you have smaller bands down here, larger bands down here, and then that's why the uncut DNA is higher in the gel. And so the Northern Lights assay is a two-dimensional, not just a one-dimensional assay. And beyond just separating based off of size and length, it's also able to separate based on other characteristics, such as whether it's single-stranded or double-stranded, or if there are other structural aspects of DNA that make it different from what you normally think of as your double helical structure. And because it can separate based off of these characteristics, it's able to both detect and differentiate between a wide variety of DNA lesions and types of DNA damage. And so how exactly does this work? So first you run it in one dimension, like the other gel that I just showed you. Typically if you're working with large DNA, and so this includes mitochondrial DNA or genomic DNA, that those DNA fragments are way too large to actually effectively migrate and go down into the gel. And so what we do is we digest it with that restriction enzyme that I mentioned before in the other gel, so that the DNA can actually go into the gel and we can work with it. And it's also inserted into the gel alongside this double-stranded marker that is at known length, so we can kind of see if there's any specific thing happening either at a certain length, or also just so we know what we're expecting to see with double-stranded DNA in the sample. And after running in this first dimension, kind of like normal, we heat treat it. And what this does is separate the DNA from double-stranded, because DNA is typically double-stranded, and it separates it so that all the DNA in the sample now becomes single-stranded. And then we run it in the perpendicular direction for the second dimension. And so if you don't have any damage in your sample, this is what you would expect to see. Again, the undigested sample is not really able to move much in the gel, but you still have the marker is able to migrate. And if you have the digested sample, you get this nice arc, and kind of this is why it's referred to as the Northern Lights assay, because typically your gel background is black actually, and so you have this nice glowing green swath across the gel, and so it really actually does look a bit like the Northern Lights. And so this is without damage. However, if there is damage in the sample, a whole lot of things can happen. I'm not going to try and go into all of this. It took me multiple weeks to fully wrap my head around this. I could spend probably 2 hours trying to explain what's happening here. But what's key to understand is that you're able to tell a bunch of different types of damage, such as if there's single-stranded DNA, if there's single-stranded breaks in the DNA. So that's when you have a break in the DNA that only goes through one strand, but not two. Sometimes you have damage which connects the strands together more with covalent bonds, which is not what typically happens. And so those are called interstrand crosslinks. And there's a few other types of damage that you're able to see, but it is a really innovative tool that was actually developed by the lab that I joined here and was the reason I came to this lab. And so I have one gel I can show you of the Northern Lights assay. And so again, like I mentioned, it does look like the Northern Lights in the sky. And this was while we still had some genomic DNA contamination that we were working with, but you have the arc of double-stranded DNA with the marker in red migrating alongside it. The sample was in green. And then what you might notice is beyond just the arc, you also have this straight diagonal line. And what that indicates is significant single-stranded DNA. We do not yet know what the source of that single-stranded DNA is, but we do hope to study further and try and figure out what the potential biological source of that is. We ran into a few challenges with this, and one of the things being the imaging machine broke. And so sadly, that's why I only have a preliminary finding and not findings with some of our better isolated samples. And some of the side projects that I then took up in place of that took a little bit more time due to the unfortunate effect of Iceland not being, a lot of companies either don't ship to Iceland or it takes a while for materials to reach Iceland, which is very unfortunate given that there's a lot of actually very interesting genetics research occurring here. And so even though I don't think I'll end up in industry, if I end up in industry, would hope to kind of maybe remediate this and make sure that try and either have a distributor here or find some way that Iceland can be better looped into these reagents being distributed so that science can occur here, not on a delayed pace. And moving forward, there are a few side projects we hope to do. Here's a picture with my mentor and a girl from the Netherlands who worked with me on the project for multiple months named Lena. some side projects we hope to do is quantify how much our protocol enriches for mitochondrial DNA over genomic DNA compared to DNA from whole blood. Conduct some sequencing of the DNA samples as another way of looking at damage. And sort of the big picture why of the project is we want to study both samples from healthy patients and samples from subjects with mitochondrial disorders, Parkinson's disease, or other neurodegenerative diseases, because these diseases are known to be associated with mitochondrial dysfunction and some types of mitochondrial damage. And it would be really informative to learn a little bit more about exactly how structural damage to DNA is related to how these diseases develop and present. And, sorry. Maybe this will go back. There we go. Okay. So now I'm gonna quickly photo dump some of the fun stuff I was able to do because I sadly don't have many pictures or lovely field pictures like Mia did. But in Reykjavik, I have just really enjoyed being here. Some of the things I want to highlight is even though I was a track runner in college, I only sprinted and did pole vault, so I've taken up more distance running along the southern shore, which has been beautiful. I've also picked up knitting. Most of these have been gifts for other people, which has been really fun. And I just, every time there's a nice blue sky, the scenic view of the city from across the pond is always one of my favorite things to see. Probably the biggest thing that was mentioned in my introduction was the dance group I was a part of called Háskóladansin, which is primarily students at the university. And I took classes in country swing, Lindy hop, West Coast swing, and then the video up top is from a contemporary class that I took. It was really a great way to meet both some Icelandic students and Icelandic residents, as well as international students from a bunch of different countries. And dance is something near and dear to my heart. And so being able to find a dance community here really made my experience, I think, what it was. And lastly, I want to just show some pictures from sightseeing. Again, you have, like Mia showed, Brewerfoss and Kirkjufell. I have some pictures from Deerley, which is honestly one of my favorite sites along the south coast. I just love the viewpoints. You have looking out on the ocean from that point of view, and you also have the view looking back at some of those lovely glaciers down there. Another picture from the Northern Lights. I had to include another one, but being able to see around this beautiful country has been probably some of my favorite travel memories in my life, and that also would not have been possible without some of the visitors I had. And so I had two of my childhood best friends came to visit me, and we did some fun activities such as snorkeling, as you see up in the left. I did glacier hikes with them, and I also had my partner come and visit, and just being able to explore, not just by myself, but with people who I've grown up with and people who are really near and dear to my heart, has really made me appreciate the country even more. And so I cannot forget to mention my family, including my parents and my brother, and it's just truly a wonderful experience to experience Iceland with the people you love, and being able to share the things that I know each of these people individually would like in the country and with everything that it has to offer. And so I just want to have a quick thank you to the friends who I lived with at my house. I lived with usually around 6 to 7 other roommates, the friends I made through Háskóladansin. Thank you to my family and friends who have come to visit me, including the ones who are currently here. Shout out to Carolina and my grandparents who are watching on Zoom. And also thank you to the Fulbright community, to the other Fulbrighters and the Fulbright Commission for all of your support and for being a built-in community that really helped me adjust to being here, and were always shoulders to lean on, and people who understood what I was going through and introduced, and being able to even do some traveling with them. But thank you so much, everyone, and I'm happy to answer any questions that anyone might have.
So we can take maybe one question or two, yes?
Can you talk to us a little bit more about the broader applications regarding why is it important to assess mitochondrial damage?
So the diseases that I mentioned, such as these known mitochondrial disorders and Parkinson's disease, There are a few types of damage that we understand are associated with these diseases, such as oxidative damage is one type, and we have a better understanding of sequence mutations. However, we know that there's structural damage to the DNA in these diseases, but we don't understand exactly how that then is related to how these diseases present, what the symptoms are of the diseases. There isn't actually, this is one of the only assays and experiments out there that allows you to look at damage in this way. And so we want to be able to better understand that. Potentially, if you're able to recognize patterns from these diseases, maybe it could be used as a diagnostic tool. If you start seeing these damage patterns in your blood, it could be a way to test the severity perhaps of these diseases. And also, if you're better able to understand the types of damage happening, you can have more specific drugs or treatments for exactly the types of damage that you're seeing in those diseases.
Right. All right, before you go, let me just present you with a certificate from
Thank
Fulbright
you.
Iceland and a little bit of swag from Fulbright Iceland. Thank you for coming and taking part of this important research. We really look forward to seeing what comes of it. So thank you so much. Woo. We really are off on a good start today, I must say. And I'm sure the next one is going to be just as great. Next up is Ben, who's a graduate of the University of Vermont. And he's been engaged in what I think is a fascinating project, researching the Icelandic Arctic fox den sites. And he's hosted by the Icelandic Institute of Natural History. Often researchers do have to pivot a bit and make adjustments, once they're diving into their work and see where their circumstances take them, and their data. And that can be a really valuable experience for a young researcher, I think. And I think that Ben has adapted extremely well here. And so I'm going to give him the floor and let him tell us all about his work.
Yes, so hi everyone, my name is Ben Simmons. I'm a wildlife biologist, and I felt very lucky to have spent my Fulbright time studying Arctic foxes, which are maybe one of Iceland's most charismatic and functionally important species. But before I kind of delve into some of my project specifics, I wanted to talk a bit about my research interests and underlying motivations for pursuing my work here. So my long-standing research interest has been community ecology, so meaning a more holistic study of ecosystems, the interactions within them and their structure. And I believe that this kind of holistic study is becoming more and more critical during a time when climate change's impacts are more and more expansively impacting ecosystems. And in addition, the systems that most interest me are those that are particularly susceptible to the impacts of climate change, those being alpine, tundra, and the subarctic ecosystems that we have in Iceland. And in addition, kind of through my previous research experiences, I formed a really deep appreciation for international collaboration. I think that in a time that we're dealing with the effects of climate change, and ecosystems are often spanning multiple political jurisdictions. Those kinds of strong global partnerships are really critical in conservation management. And so that's kind of what led me to pursue Fulbright as a means of bridging scientific research and developing those international collaborations. So I've been lucky to have this kind of balance of two hosts during my time here in Iceland. So the University of Iceland, and then also the Icelandic Institute of Natural History, which for maybe Americans who are not familiar is a government organization and is equivalent-ish to Fish and Wildlife Service. And that's kind of where I've spent the bulk of my time. I've been really lucky to have found a community there where I can kind of intermix with people that are pursuing projects adjacent to my own. And I've had the opportunity to hop on a lot of their projects as well. So I've been able to go to a lot of different parts of the country and help out with bird surveys to study how the bird communities are changing in Iceland. And I'm going to be able to help with gyrfalcon nest monitoring and ptarmigan surveys as well. during the remainder of my time here. And what I'm most excited for is going up to Hornstrander at the end of June and helping out with fox den monitoring and hopefully seeing some pups. So. And it was also within this work community that I was kind of struck by one of the biggest cultural differences that I think that I was able to see, which was that it felt like because of a smaller population size compared to the US, that it kind of sometimes results in this feeling of greater personal responsibility. And I saw people chipping in and stepping up to help others with projects in ways that maybe I wouldn't have seen as much in the US. And also I was able to see that citizen science was maybe more a popular method and also maybe more effective in Iceland. And so that was something that was really cool to see. In addition to my host institutions, I was also a member of an international Arctic fox research team. And this is kind of where I received the bulk of the guidance and the support for my project. And it was also through this team that I was given the opportunity to go up to the West Fjords and participate in some of their field work, but also meet with fox hunters and some other stakeholders in fox conservation. And this team, though relatively small, is made up of 6 nationalities, Iceland and the US included. And so it was just something that kind of has further stoked my desire to be involved with international partnerships, as it's just yet another example for me of how effective that can be. And so transitioning into talking a bit more about my project, Arctic foxes are a species that I wanted to work with for a really long time. And as a community ecologist, there's a few reasons that they're a very interesting focal species. So they have a really expansive range, and throughout most parts of their range, they're a keystone species, meaning that they have a disproportionate impact upon their ecosystem relative to their population size. And in fact, when studying the Arctic, they were even named a flagship species for studying climate change by the International Union for the Conservation of Nature. So by studying them, we can almost indirectly monitor the impacts of climate change upon their entire system. And like I said, you can see that they have quite an expansive range, circumpolar. And so, but in addition to their more innate characteristics, the Icelandic population offers an even more interesting focal population. They're Iceland's only natural terrestrial mammal, so those keystone impacts are maybe even more enhanced in Iceland. And then also with regards to studying climate change, Iceland is warming at a rate that greatly exceeds the global average. And so by studying here, the effects are maybe even more apparent, and then also it could serve as a look into the future of other regions. In addition, unlike other parts of Europe, maybe, where the fox is really struggling, the population in Iceland is actually doing pretty well, and it has actually 90% of the Nordic population. And with one of the highest fox densities up in the West Fjords in the world. So, but also Iceland is very distinct ecologically from other parts of the fox's range. And so there are a lot of unanswered questions here because tools that have been developed in other parts of the fox's range are less effective at being used in Iceland. And another key difference between Iceland and other parts of the fox's range is that hunting pressure is a major source of fox mortality in Iceland. And fox hunting has pretty much taken place as long as humans have been in Iceland. It was actually written into the first laws here in 1295. And though this is a major source of fox mortality, it's also developed a very unique and expansive data set. So there were collaborations that have kind of been formed since 1978 between fox hunters and the Icelandic Institute of Natural History. And through that data, through them, we can get a lot of data. So they send in carcasses and information about where it was killed, its breeding status, as well as locations of known den sites. And by dissecting and doing other things, we can get a lot of data from that. So you can see me here scrubbing muscle tissue off of a fox head after it's been boiled for several hours to soften it up. But through that kind of gnarly activity, we can extract things like teeth and age them, and also see their reproductive capacity, we can conduct a genetic study, we can see what their diets are, we can do morphological study. And then there are also, now we have accumulated this massive data set of known den locations, over 8,500. And that data is kind of what has become the foundation of my work here in Iceland. So those den locations my overall goal was to hopefully use those den locations as a proxy for what is suitable habitat for the foxes. So my overall goal was to create a tool that could be used in conservation management in Iceland, as this fox data hadn't necessarily been, or this den data hadn't been spatially analyzed, and especially not at this scale before. So specifically, my 2 research questions were what habitat conditions help determine suitability for dens, and then how may den distribution change with climate change? And so to accomplish those goals, I used a method called species distribution modeling, which can predict where a species is likely to occur based on presence data and environmental variables at those locations. And this can produce a lot of really interesting results that are pretty key in conservation management. And so I've tinkered with a lot of environmental variables. But the ones on the screen are those that I've found to be most significant thus far. You can see this is the maximum temperature averages of the hottest month with hotter temperatures in red. And this is what the variables look like when they get put in the model. So that maximum temperature was chosen as obviously foxes are a cold adapted species, and so we predicted warmer temperatures would negatively affect them. The precipitation for the month of June was selected because during June, foxes are in a really energy intensive period. They're raising their young. And so getting insight into the weather conditions during that time. Elevation makes its way into a lot of these models, as it tells you a lot about the species ecology, the plants, the temperature, the snow cover, things like that. The slope of areas was included because anecdotally, we know that foxes in Iceland select moderately sloped areas in order to get protection from the wind, we think, as well as there's more rocky cavities for them to find a den in. And then also the distance from roads, as roads can be potentially a source of food, like carrion and like roadkill, and also human food waste, but also they can be a source of disturbance and mortality. And then distance to coast, as Icelandic Arctic fox diet has a lot of marine resources in it, and that's something that they really depend upon here. So this is a result of one of the models, and so the cooler temperatures are indicating lower habitat suitability, while warmer colors are indicating predicted high suitability for the fox. And so the goal of this model, and I should mention that this is using all of the variables that I mentioned with the exception of the maximum temperature. So that'll be in the later model. But the goal of this model was to match the known distribution. So those den points I showed earlier, as well as possible, and also to highlight areas that are potentially not surveyed by hunters, but that may be overlooked quality habitat. And so if we take that exact same model that was on the previous slide and just cut it down to the threshold that's in orange of what the model predicted to be suitable habitat, and then we overlay those fox endpoints, we can see that it's matching up pretty well. So I was pretty happy with this. One limitation to note of this model is that you can see these little points in the middle here. We know there are foxes in the highlands. There are less foxes in the highlands, but they are there. But also there's less hunting there, so we don't get as much data from the highlands. And so this model will be inherently a little bit biased to being able to be predicting in more coastal regions. And to begin to answer my second research question on how climate change may impact distribution, I've been working on this model that uses the same covariates, but replaces that elevation with the maximum temperature of the hottest month. So since all of the variables that I used are abiotic, they're things that are not going to change that much in the future. Elevation is not going to change that much in the future. with the exception of those two climatic variables, precipitation and temperature. So we can create other models to compare to this one. This is modern day, and just exchange those two climatic variables with the predicted values for new in the future. So obviously predicting into the future deserves a major disclaimer, as this isn't, this is predicting out pretty far, and this isn't going to say what is going to happen, but it does give us clues on how environmental conditions may affect suitable habitat in the future. So we can take that same model on the previous slide with modern day, and then this is a forecast model for the years 2041 to 2070. And you can see that the suitable habitat has kind of shifted northwards. And maybe there's a slight growth in the north as well. And then even we can predict out to 2071 to 2100. And so now you've seen that it's moving further into the east and the north is even further. And this time resulting in more shrinkage. So there may be concern that it's shrinking up northwards. However, moving forward with my work, I will be working to refine this model further and the other model and experiment with other climatic environmental variables that we can include that may give us more information, as well as find better ways to predict how warming temperatures will affect fox habitat distribution, because we have so little data on how this cold adapted species will react to these projected temperatures. So moving forward, I'll continue to refine this model and work to publish the results. And then this summer, I will be starting a master's in polar studies at Cambridge, working with Greenlandic caribou and muskoxen. And I'll be using many of the skills that I've developed during this fellowship in that work, and hopefully continuing to cultivate international collaboration, hopefully to see some foxes in Greenland this summer. So thank you so much.
Fascinating, yes, Pat.
Can this kind of modeling also take into account human variables? So like in Hornstrandir, there's nobody living there.
Right.
Or for example, like connection or proximity to eider farms, because if they're being shot because they're close to eider farms, then wouldn't the pressure be more there? Or, you know, like how can you build in some of the like human factors in models like this?
Yes, so there are a lot of variables like that I have included, like distance to buildings, for instance, or how far they are from. We have a map of the shooting hides that hunters use. So like, are they avoiding areas that there is a lot of hunting activity? Or even like prey availability, like we have maps of like where the pink-footed geese or the ptarmigan are predicted to be. And so those can be included as well. Those didn't end up being significant to include in the first model that I showed, but I am still playing with ideas of variables like that in that model. But obviously I wouldn't want to include variables like that in something that I'm going to predict into the future, because it's hard because with climate variables, we have data on that we can project into the future, but not for things like that necessarily. Yeah.
Thank you.
Yeah.
Yeah, let's hear. Okay, over here, Julio.
So then models are always changing as you adapt them.
Sure.
I'm curious about the variables that you have and how you considered weighting them in different ways.
Yeah, so I mean, I didn't get a chance to talk about this, but there are some things that I played with different variables. So like some of them are getting treated linearly, while things like slope, where we have this really clear optimum, like they're really choosing things on a moderate slope, that's getting squared as well. So there is a squared slope variable along with the slope variable. Or things like, I forgot to mention this, but one of the models included an interaction term between the distance to the coast and the slope, because obviously, like, if they are preferring areas close to the slope, but then Iceland has these coastlines that are sometimes just a cliff, they're not going to den there. And so we want that interaction as well. Yeah.
Yes, Carrie?
Quick question, do trees matter at all?
I did play with NDVI, which is a way of looking at vegetation, but the elevation or the temperature variables are so strongly correlated with that you can't include them without overfitting the model. And elevation and temperature were the things that ended up being more significant, so vegetation didn't end up making the cut.
We've got back here, yeah.
I wanted to say, first of all, there's a very good map of Eider farms, if you're interested.
Okay, yeah.
If you haven't already found that. And then I was just curious what specific model you used, like juice and cheese distribution model.
Yeah, this was done in R using a GLM, generalized linear model. Yeah.
It was somewhere, yeah. Arisha.
You said that Arctic foxes had like an overwhelming impact on the community that they live in. I was just like wondering like what makes them have such a huge impact on it.
It's in a lot of ways. So I mean, here in Iceland, they're the only like terrestrial apex predator. So apex predators are always going to have a big impact on the rest of their system because everything that they eat, they're influencing that population and thus everything below them in the food chain. So that kind of cascading effect is innate to apex predators, and then that effect is even greater in Iceland, where they're the only terrestrial apex predator.
Okay, we've got one final question online.
Yes, this is from Zoom. It asks, I'm curious if in your research you found any information on how human interaction, i.e. people visiting this location, feeding the foxes or leaving food, refuse behind, is affecting where they roam, or if anyone has looked into that.
There is some work going on from my mentor, Esther, on the impacts of tourism, especially up in Hornstander. That's not something that I was able to easily include in my modeling, as there isn't, if I want to model for the entire country, there isn't that kind of nationwide data on tourist interactions.
Okay. Good, fascinating. Thank you so much. We can give you a certificate of completion, a Fulbright pen, and a little Fulbright memento.
Thank you.
So best of luck with your studies and your trip to Greenland over the summer, and then your studies. We look forward to following you in the future.
Thank you.
This is also interesting. Yeah, so we have a final speaker before the break, and that's Melissa, who comes to us from Brooklyn. She spent her time in Iceland working on a unique independent research project at the Reykjavik School of Visual Arts, and was also hosted by the University of Iceland. It's so important for us to have artists among our Fulbright grantees, and we have been really excited to support this project on exploring sustainability practices in textile printing and production. So we cannot wait to hear from you.
Mine's gonna be a bit different from what you've just seen. Maybe a lot of pictures in process. But yes, my name's Melissa. I'm a printmaker and I'm an illustrator from New York City. Recently have been also dabbling in teaching. I've been in fine arts and production for the past 10 years. And so my goal is coming to Iceland. I really wanted to meet artists, especially textile artists, and seeing how they were implementing natural resources and dyeing and what sort of things were available in Iceland. I really wanted to learn about dyeing. Knitting is also something that is very unique, or it's a unique environment to learn it in Iceland. It's a really core part of the textile culture here in Iceland. History of textiles, foraging, looking at businesses who are implementing sustainability, and of course my practice of print and how I can implement natural dyeing into my print practice. And just in the bottom corner, I thought it would be interesting. This was one of my dye pots that I had left for about like 3 weeks, and it built this beautiful colony of mold, and I thought it was grotesquely beautiful. So when I first got here, I started to just look into textile artists, and I was cold reaching out to a lot of them, seeing if they would talk to me, invite me to their studios. And a lot of the artists that I was talking to in Reykjavik told me about Gudrun. She has a studio in Selfoss. And she is mostly doing everything with natural dyes, actually is. I think she's also a botanist. And so she invited me down. They had Wool Week in October. And I went down there to do a workshop with her in indigo dyeing. She mostly dyes wool. And you might see her wool around Reykjavik. I think it's really unique. Pretty much she will just dye with stuff that she finds around. And if she doesn't like the way it looks, she does this tie-dye process with the indigo and will dip the yarn in indigo, and you sort of get this tie-dye effect, which I thought was really, really cool. And then another textile artist that I linked up with was Kristalina. She is a Fulbright alumni, and she's here in Iceland teaching about natural dyeing. And I did 2 workshops with her, one with just basic natural dyeing, and then I also did an eco-printing dyeing workshop with her. So these started my foundations for dyeing. I had never done it before. There's a lot of chemistry behind it. And so I embarked on combining the 2. So I mostly was doing screen printing, and screen printing is a stencil process. You have an image and you're pulling material through it onto a surface. In this case, I was mostly just doing fabric. And I worked with these 2 print pastes, alum paste and iron paste. The alum, it basically helps prepare the fibers to chemically bond with the dye. Without a mordant, it will dye the fabric, but it'll wash out over time. And then also iron is used as a mordant, but it creates a strong bond, but it also does what they call in the industry, it saddens the colors, so it makes them darker. So these were the 2 pastes I mostly worked with. And so this was my very first round. And I wanted to show you like what my film setup looked like. I wanted to test to see like the complexity of an image I could get, how thin of typography I could print, and then like what just like a graphic image would look like. And then this, you can sort of see it. This was one of my first prints with the print paste, and I started to realize early on like the print paste just, it's kind of clear and I couldn't see like my print quality. And this was my first result. It worked, but it isn't perfect. I had a lot of noise in the background. I was actually quite surprised how clear the typography came out, but as you can see, like the images, you can't really tell what they are. And so I experimented with more print paste. This was sort of my setup at Lista Scoli. And in this case, I added a bit of dye to my print paste so that I could see the image quality. And just recently, this was probably the best run of prints I got. And I changed up my formulas for the print paste because the other ones I was working with were not working necessarily. So this one, I worked with that my liquid was mostly distilled white vinegar and little water. And this was a 2-color print. I wanted to achieve getting 2 tones within one image when I dye it. So as you can see on the left, I freshly printed the iron paste, and then as it sat on the fabric, it started oxidizing, which I thought was cool looking. And then I wanted you to see, I love doing this technique in screen printing. It's called a blend, where you blend colors together. And I wanted to see how the iron paste and the alum paste would interact with each other, and if I could actually get a blend. So this is what it looks like when I first printed it. And how I also changed up my process, I was reading into doing this fixative bath before putting it in the dye, which it's called a dung bath, but it's not made with dung. It's made with calcium carbonate and wheat bran. And so it's supposed to help fix the mordant into the fabric and also have that like the dye doesn't attract to the areas I don't want it to print or to attract to. And what's nice about this is if you test the pH of it, you can just like water your garden with it. It's not like a toxic thing. And so this was really cool. This is a dye bath of just tannins. And it's really cool to see how the iron interacted with it almost immediately. It immediately sat in the color. And I thought that that was so cool. I wanted you to see a video of it. Oops. No.
Is that the video?
It is the video.
There should come up in arrows.
Oh, yeah, go to another website for the video.
Okay. Sorry. Anyway, you can sort of see it from this photo, the previous photo, the color changing. And these were the final, after they dried the next day. I was really, really happy with these results. And also the fact that I even got that crisp of a line in those really small details. I was ecstatic with these results. And I'm going to continue. I'm going to be still staying for the next 2 months. And one of my goals is to go foraging. I want to use some more Icelandic. Obviously with the weather, like in the next, I think by the end of this month into June, there's going to be more options for me to forage. So I'm trying to find some Icelanders to take me foraging. Yeah. And so at Myndlistarskóli, I really wanted to give back to the students. I wanted to teach some printmaking. And I worked with Margaret, the head of Textiles, to come up with this block printing workshop for the students. I tailored the workshop in a way that it would be accessible to them without a studio. So I built it around just being able to, as long as they had a matrix, which is the linoleum or rubber to carve into, paper or fabrics. And then I used very like sort of household items that you could use for carving. And so we covered the advantages and the disadvantages of the two mediums for printing. And it was really well received. They really liked it. This one girl on the left, Hannah, she went crazy and she was like hand painting on them and just like, she just kept going. It was really, really fun. And even Margaret told me after the workshop that she said like they were so focused. She's like, all semester, she was like, I didn't see them that focused before. And it was really great. They had a ton of fun. And I think it was a nice break from like their projects for the semester. And this is just some more work. That came out really, really great. And then knitting. So knitting is, I think in Iceland, it was probably one of the coolest things to unlock in Iceland. And I'm going to preface, I've never knit before coming to Iceland. So it was really great for me to connect with other Icelandic knitters. I went to like the knitting group at the library. I also would go to these craft nights at another boutique. And I got to talk with Icelanders about and just get knitting tips. And so these were some of the first projects I worked on. I started small with dishcloths and then hand warmers and hats and this thing on the top. And then, so to prepare myself, I really wanted to learn lopapessa. I didn't know how difficult that was going to be. I really thought it was like a far-reaching goal for me. So for that, I like to make learning fun for myself. So get ready for the next slide. I knit my cat an Icelandic sweater, or at least an Icelandic wool sweater.
I have to commission one.
Yes, totally. I thought it was fair to put it in here because as much work as I put into making it, getting it on him was also just the same amount of work. And I thought he looked so cute. And this was about like 10 minutes and then he was like, I'm over it. Anyway, so that sort of set me up for like, I've never like knit any sort of garment, but it was sort of like small scale, low risk. I felt like, you know, to start me off. And then, so this was my first lopapessa. I followed a knitting pattern from Helena Magnusson, who is also known as the Icelandic knitter. She does these knitting tours near Selfoss. And I followed her pattern because me as an illustrator, and I also really gravitate towards florals. I really liked this pattern. It was called like My Icelandic Sweater, and it was based off of traditional embroideries, I think, for like certain shoes and gloves. As you can see, for my first one, I mean, it's a sweater, but I knit the color work. It was my first time ever doing stranded color work, and I knit it so tight that it doesn't even really fit me. So I learned a lot from that, and I was like, okay, I think I can do this. So I decided to embark on designing my own. So I went and looked through books just to see like the analog process of like how they construct the design. This was inspiration. I took photos of the embroidery at like the National Museum. And so I took inspiration from this. And on the left was my initial sort of digital sketch. I was like, okay, here's the design. And then I realized it was way too complex. So I decided to go analog. I'm more comfortable going analog. And as you can see, I'm sort of referencing the other pattern. I sort of backwards engineered that pattern just to figure out where decreases happen. I felt like that was the hardest part. And I wanted my sweater to be a little more original to the original loaf of pizza. I didn't realize that it's actually a pretty new commodity. They started making them in like the 1950s. And originally they would have a yoke design and then simpler elements from the yoke design on the cuffs and the hem. But in 21st century, Icelandic knitters are pumping these out really, really fast. They simplify the designs. So most of the time you only see the yoke. Because all Icelandic sweaters are hand knit. I actually learned the Hand Knitters Association, I think employs about 500 knitters to knit sweaters, which I was like, damn, that's a lot. And so here's my sweater.
Wow.
I designed this sweater, and I'm really, really proud of it. I did not think this was gonna be achievable in this time, but you know, the winter months are dark, and I did a lot of knitting in winter, and you can see the difference between the two sweaters. And I tested it out on a hike, and it is definitely a very functional piece of clothing. The sheep here have developed coats that can withstand the environment in Iceland. So the wool here, they usually spin both the top and the bottom coat, which is why it's kind of itchy. But it's really functional in that it is waterproof and wind blocking, actually. Anyway, I had some side quests besides the textiles. I came to Iceland 2 years ago, and I immediately felt how creatively charged Iceland is, and I was so inspired by it, especially the landscape and all the textures. I feel like every time I went out into the wilderness, I have been staring at a lot of mountains and the landscape, and I started to do a series of sketches, and I'm mostly looking at maybe things that like other people are not necessarily looking at. And I've done about like 20 of these sketches, but I just decided to highlight these four because I turned them into drawings, which then I transferred to blocks. And I started making these block prints after I did my block printing workshop. I only brought 4 of this size. So I only did 4 of them, but I want to continue this series. I kind of want to blow them up to like 18 by 24 block prints. And these were some of them printed. I actually really like these images, and I think that they translated quite nicely to block prints. And then this is the wall of my bedroom right now. I've also been working on a graphic novel, which I'm pretty much only in like the story phase. So I'm just plotting a lot of like plot points, and I plan to use Icelandic landscape in some of the drawings and some Icelandic folklore as like archetypes of characters. And these are just some preliminary sketches or just like concept sketches I started. It was going to be a sci-fi fantasy novel that I'm probably going to work on for many years after this, but definitely inspired by Iceland. And then fun friends culture in Iceland. I had a lot of people come visit me, especially my parents, my family. It was really exciting for me to bring them. I love that you guys eat ice cream any time of year. It doesn't matter the temperature. I'm all about it. I have my parents. I think that was like, I think it was like, it was really cold, and I got them to eat ice cream. But yeah, and also, you know, my fellow Fulbrighters, I feel like I've been completely inspired by all of you and all of you sharing your projects and your knowledge. And I feel like I've become sort of a geology enthusiast at this point. I love rocks. And yeah, you know, I also made some artist friends here, and I went to some of their openings, which was really cool to also meet other artists. And then also the craft night I kept going to. Sigrun is my friend now. And that's sort of been a core part of me, you know, meeting other Icelanders and also other expats who have been here. And we're all just sitting there doing crafts together. And I learned a lot of cultural things. And even at one of those nights, which I thought was like one of the coolest things, I met a woman, And she was sitting there, she was just spinning some wool. And I was like, oh, I was like, what is this contraption she was using? And like, I was just asking her, she's like, oh yeah. She's like, this is from a lamb that she's like, I help birth. And now I'm making yarn from the wool. And I was like, what? Like, I'm just like, to have that, to just have that connection with the material and like where it comes from, I feel like is something that I've experienced over and over again in Iceland. And I just thought she was so cool, and she was just like, yeah, it was just like another day for her, which is really cool. And then of course Iceland, you know, I mean, I'm also a Northern Lights snob now, and just constantly, just never-ending inspiration for me. And I feel like Fulbright and being here in the context of Fulbright has been a way for me to really connect with people, connect with artists. I'm really hoping I've made connections, lifelong friends, and I really want to come back. I'm working on trying to come back as visiting faculty to teach printmaking, because I feel like I could contribute to the printmaking scene here in Iceland. But yeah, I really want to thank all of these people, and also the cats of Reykjavik, because they helped get me through missing my fur baby back home. But yeah, all of you inspire me. Iceland inspires me, and you know, you're not gonna get rid of me that easily. I'm gonna be coming back for years and years. Thank you.
Okay. A completely different topic, just as fabulous as the others. Do we have questions? First, I just want to say, can I commission a sweater for?
Of course, I will make a sweater.
For Rusina, yes.
Just gotta get some measurements.
Yes.
We can buy those block prints too.
Yeah.
Yeah,
Yes.
100%. I plan to make an edition of that.
And I was gonna say, for the commission here, it would be very nice for us to get one of those as sort of our memento from this year,
Of course.
because they're beautiful. Yes, yeah, I'm just wondering, because we haven't talked about that yet with any of the grantees, moving to Iceland, sort of becoming, what are some of the challenges you faced, and how did you face any challenges?
I mean, I faced challenges with my host institution, Liza Skoli. They were in the process of moving, and they're still settling into their new space. So, you know, there's a lot of construction going on, and there was moments where like I got kicked out of the classroom I was using because they needed it for the students, and like, you know, I totally respected and understood that. But like, I think the challenges, like, they just sort of they pushed me in other directions. Like, I think I was too rigid in thinking of what I was going to do here, that like, I kind of just feel like Iceland, at least something I learned in Iceland is like, when I don't go looking for things, Iceland just like delivers them to me. So like, even though like the challenges in themselves, I just was like learning new things, and it pushed me also to go way out of my comfort zone.
Yeah, exactly, yeah. All right, well, thank you so much, Melissa. We
Thank
are
you.
so thrilled to have you amongst our Fulbright cohort for this year. And here's your certificate of completion, and a little Fulbright memento, and a Fulbright Iceland pin. We look so forward to seeing more of your artwork, right? And seeing you here often in Iceland.
Yes, definitely coming back.
So thank you so much. So welcome to our second session of the day. Welcome back if you were with us earlier this afternoon. In the first part of our program, we heard from 4 Fulbright Fellows in diverse areas like printmaking, geology, zoology, biology, right? All of our speakers in the second segment are here on Fulbright Iceland National Science Foundation grants, but they also are engaged in very diverse projects. Although these projects do have in common that they are in some way focused on the Arctic. And I'm sure we're all excited to hear what they've been up to. So the first speaker after the break is Fulbright Fellow Jack Ivie, who is a graduate of Drexel University, and he came to Iceland with the goal of studying Icelandic forests and creating an afforestation map. Hosted by the University of Iceland, and I know that he has taken the opportunity to engage with local scholars, including Fulbright alums, and I think his work is going to provide new insights that will be valuable to the Forestry Service. So take it away, Jack.
All right, I'm just gonna jump straight into this. So I'm Jack, and I'm here to present my project, which is an Afforestation suitability map. And so I had three main goals during the grantee project. The format is off, that's gross. So it was to create first a large-scale suitability map of Iceland for birch planting projects. The second thing was to conduct a literature review of published maps, models, and estimates of the settlement era of Iceland's forest coverage, and then to use a combination of these two things, the best published model and my created map, to create figures and tools that non-ecologists could use to potentially make more informed and ethical tree planting decisions for restoration and soil remediation in Iceland. So the study species for my birch tree, study species of this project was the birch tree, is the single forest forming tree species in Iceland. It's not the only native tree to Iceland, but it is the one that makes up 90% of the trees in a forest. The size of these trees varies widely throughout Iceland. I have some smaller trees and some larger trees here that I've taken photos of, but the main size is 3 to 5 meters tall. And recently there was a publication published just last year that was a groundbreaking study in natural colonization of birch trees in Iceland. And it was the very first to compare birch colonization across all of Iceland. And so this publication was very important throughout this entire research project. And so the other thing that was a big part of this research project was a focus on the landnam era, the settlement era of Iceland. And why this is important is it provides a historical baseline for ecological restoration. And so a baseline is an environmental state of an ecosystem during the past, and researchers and restorers use these types of things to restore an ecosystem to the pre-degraded state of natural climax communities to avoid using instead what we think is the human created ideal of an ecosystem, what used to be there in the past. And why I picked the landnam era is through literature review I did before I was even accepted to the Fulbright project. It's been very heavily found and supported through various research studies and publications that during the landnam era, you see a decoupling of things like erosion patterns and vegetation coverage from natural controls like climate change. And the main culprit that researchers point to is human settlement, specifically livestock grazing, and majority sheep are the main culprit that people tend to blame for changes in these things. So I did my literature review of every forest landnam model I could find that's been published for Iceland. And the best one possible that I could find was published by my affiliate, Dr. Randvik Olafsdottir, in 2001. Every other model of landnam era forest coverage in Iceland only uses typically one variable. It's either elevation or temperature. The model that they published used various variables. They used these things called degree days, which are days during the year which are warm enough for photosynthesis, which is the only parts of the year that trees can actually grow. They used topography. They didn't just use elevation. They used various variables to create more complex models of topography. They checked their model against the published fossil pollen presence at the time. No other model has actually used fossil pollen, actual evidence of where birch was at the time. And they checked their model against modern forest coverage. And just to give like a quick representation of how complex this model is, like this is all of the variables and inputs and outputs they're using. I do not have time to explain how complex this is, but it just gives kind of a justification of how much more and complex and informed this model is than the other ones. And so this is the spatial data I created from their model. The light green areas are total vegetation coverage of Iceland. So that includes shrub birch, which is not the focus of this study. The dark green areas with the dots, that includes tree coverage during the landnam area. So these are historical areas of Iceland that used to include trees, but this is nowhere near the amount of tree coverage there is in Iceland now. There's roughly 1/10 of this tree coverage in Iceland now. And so the other main publication, which I mentioned earlier, was the big study they did on natural birch colonization. And so what they did is they found statistical significance of environmental variables on natural birch tree reforestation. And so they went to all these different study sites around Iceland where there's current old growth birch forest, and they checked out these different types of variables, and they used two main categories. They used microsite variables, which is like the surface coverage of the ground. So it could be the different kinds of bare earth, whether it's dirt or groundwool, or the various kinds of dominant vegetation. So that could be moss, grasses, dwarf shrubs. And then they had explanatory variables, which are various just different kinds of variables. They could be environmental, anthropogenic. So they included like sheep grazing, temperature, elevation, those sorts of things. And then they did statistical analyses to determine if they were significant, negative, positive, and how significant they were. And so I took the significant variables and I went and I looked for data sources where I could get data on these variables for all of Iceland, because they only had data for these very specific points in Iceland. But I wanted to create a map for all of Iceland. So I went and I got sheep grazing data. However, there is no sheep grazing data for Iceland because sheep free roam graze, and people don't exactly stick GPS collars on the sheep or all thousands of the sheep and track where they all go. So for stand-in for sheep grazing data, I calculated the sheep population density per each municipality in Iceland and used that as a stand-in, assuming that if there's more sheep in a municipality, there's going to be more intensive grazing. And this is a negative variable because very obviously sheep will eat plants. It's that simple. The largest data set that I used was a terrestrial habitat classification data set that was published by the Icelandic Institute of Natural History, and it was most recently updated in 2024. And they classified Iceland into around 74 ecotypes, and they created these data sets of each ecotype. And this contained the vast majority of data that I used. So it included the microsite data. So it had these top 10 most prevalent plant species. And so I would go through and I would look at the top 10 plant species and I would categorize them into the microsites. So for example, if it's moss, I would figure out which of these are moss of the correct type, add up the percentages, and that's how much of that microsite is present in the ecotype. So there can be more than one microsite in an ecotype. It also tells you if there's a presence of trees. This is the data for birch forests. So you can see Birki here is birch, and it's by far the most prevalent plant here. And then you have all the soil data you might need, pH, carbon percent, wetness of the soil, depth of the soil is all here. And so vast majority of data was available from here. And they had this spatial data that was down to 5 by 5 meter pixels for all of Iceland. And they got this data through over 1,000 transect surveys across Iceland and then remote sensing data as well. And so elevation data was very important. So I downloaded the digital elevation model of Iceland from the National Land Survey, and I removed 600 meters above elevation because that's considered the typical physiological limit of the tree form of birch in Iceland. And I didn't want to skew my model by mapping areas that I just knew would be completely useless for tree planting projects. Other areas are removed for like urban areas, agricultural areas, glaciers, geothermal areas, lakes, rivers, that kind of thing. No need to include them in a birch tree planting model as they're just completely unsuitable for birch tree planting. And then I added a variable that was not tested by Bridget et al. Study, and this was human disturbance data. I weighted this the same as the sheep grazing variable that they tested because it is another disturbance variable of an animal trampling that weighs over 100 pounds. And so I went to the website Strava, which is a web service that tracks exercise, and I took heat maps of foot traffic data, I screenshotted them, I geo-referenced them, and then I turned them into spatial data that gives you an idea of human presence outside of urban areas. So for example, this is the Reykjavik area. That all just gets deleted because it's an urban area, it's unmapped. But all these other areas out here that are kind of out in the more wilderness area, that is all then included in the later map. And so how I implemented all this data into the map is I used ArcGIS Pro, a geoprocessing tool called Weighted Sum. As here's a demonstration of pretty simple demonstration of how it works. You have two variables, you have your pixels where all your data is on. You input your data. I put all of the variables on the same scale of 1 to 20 because you don't want to be comparing apples to oranges, obviously. Like if you have elevation that goes up to 600 and you have a percent that only goes up to 100, you just cannot just add those together. And then you get the weightings. The weightings applied by the computer to the data, and then it outputs them and you get a variable. And so the weighting I estimated was used from Bridget et al. This is all the variables they tested. Only the colored variables here of these microsite types are significant. The green ones are positive, the red ones are negative, and then here only the dark black variables are statistically significant. And so once you input this all in, I get a suitability map of Iceland for potential birch forestation. And so I've colorized it from most suitable to just completely unsuitable for birch forestation. And so each of these pixels here is a 5 by 5 meter pixel, so you can really zoom in to like local areas and try to find specific areas for birch planting. I'm going to kind of give a demonstration of how I want these tools to be used by non-scientists when they're trying to figure out when they want to go plant trees. So before I had done any mapping, I wanted to kind of hypothesize where I would think, if I was a stakeholder, I would want to plant trees. So I had two areas I picked. Over here is the International Airport area, and I figured that this would be an obvious place to encourage some forest planting, as there would be international tourists coming through here. There's pilots and things like that. There's airport staff that might be staying in the area, and maybe they want to engage in some ecotourism. I'm also in the tourism department with my affiliate Randberg Olafsdottir, so ecotourism is going to be a big part of the publication that is eventually done. And then the other area over here is an area by Hypothermic, the Glacial World Guest House, and there's other guest houses around here, so it's kind of another tourism area. And so I mapped out the historic forest coverage, and both of these areas fell within historic forest coverage, so that's a plus. I also mapped out these barren land cover types where there's very low vegetation coverage, so low vegetation to compete with the trees. So these areas would potentially be good for trees because there's nothing to compete with them. And then I also mapped current vegetation, forest. So pink is current birch forest and orange is current forest plantations. And so if there's already current forest existing here, it stands to reason that this area is probably going to be pretty good habitat for birch tree. But I wanted to see if my model actually bared that out, if it lined up with my hypothesis. And so go to the next slide, and we can see pretty clearly that at least this area down here where there was the bare vegetation is very well suited for potential tree planting. The area around the airport here, you can actually see the airport's like runways and stuff here. There's a lot of really good areas here. Remember that each of these pixels is 5 by 5 meters, so you could really zoom in here and try to plant in these little areas here. The areas around the guest houses here, these big red blotches, that's actually current forest coverage, because one of the things that they found in their study, and if it makes logical sense, that if you have current forest, you're not going to be able to go there and plant more trees. They're going to be unable to compete with the forest that already exists there. But you go just next to that forest coverage, and the habitat is very similar. It is very suitable to the already occurring forest that exists there. And so this is how I would, in theory, want somebody to use these maps and figures, is to combine the historic coverage of forest with my suitability map. I would not want people to plant forest outside of where forest used to exist. We want to conserve other ecosystem types in Iceland that are important for other species that don't live in forests. That's that slide. I think that's it. Yeah, no, that was it. But yeah, so what's the next steps with this data and these figures is I'm going to publish a manuscript of my methods and the use of the map, where I got all my data from, and I'm going to publish it in two sources that are associated with both my affiliates. One of my affiliates, Dr. Randberg Olafsdottir at the University of Iceland, has published previously in the Journal of Arctic Tourism, and so I'm going to publish my data there, and we're going to have a bit of a focus on ecotourism and how you could encourage reforestation and ecotourism in Iceland with natural birch forest, kind of sell it as the real Iceland kind of thing. And then you have the Wildlands Research Institute at the University of Leeds, which my other affiliate, Dr. Stephen Carver, is the director of, and he's worked previously with other mapping projects in Iceland. And so we would we would put the publication on his website as well, along with links to the raw data itself, so people could go and open it up and zoom around the map of Iceland and get really down into the 5 by 5 pixel areas and look around. And if these people know ecology, they know GIS, then they can really get in and look at my models and see if they agree with the way they're done. One of the things that I wanted to mention specifically is that a model is not a substitution for going in and doing ground truthing. A lot of this data was done from remote sensing from either the air or from space. And I used all the data from the Icelandic National Institute is the average of these ecotypes across Iceland. So in some of these areas, the pH might be higher or lower, or it might be drier or wetter than the average. And so that means that it might not actually be as suitable for birch trees as you were thinking, or you might get there and there might just be a bunch of rocks there instead of dirt. And that might have not shown up on the remote sensing data once you get down below the 1 centimeter of moss that's there. So there is limitations to the model. And so ground truthing should always be accomplished, should always be accompanied with any kind of planting project, any kind of models like this. And then I also want to add wind data from the Icelandic Meteorological Society. Once they finish their website data portal, I contacted them at the beginning of the project, and they told me to wait till the data portal was finished. And then I contacted them at the end of the project, and they told me to wait till the data portal was finished. So that wind data, the wind variable was found by Bridget to be very significant for birch trees, as birch seeds are spread by wind. So if you want to plant trees in sort of core areas and then the wind fills in the areas between them, you're going to save money and time instead of planting across the whole area. And so that is kind of one of the things I want to do in the future is add more variables, maybe also add things like aspect and slope to make the elevation and topography part of the map more in depth. But that is essentially the end of the science part of my presentation. I have some photos here of fun things I did while I was in Iceland. I really got into like the holiday spirit in Iceland. I really loved the pagan feel of it, like the trolls and the monsters and that kind of thing. I really enjoyed that. I joined the gaming university club in Iceland, met a lot of Icelanders and international students like that. These are two of my friends from Texas that I brought down here, or up here. And one of them had never left the country before. So this was a pretty, I think, unique experience for them. And then I made friends with some Icelanders. This is one of my Icelandic friends. I was at a concert here, and I really enjoyed Icelandic cuisine. I really enjoyed trying all the, I guess you call it unique foods. And yeah, I loved all the Icelandic holidays and all that kind of sort of thing. And so I wanted to take some time to thank a couple of organizations and people. Obviously, my two affiliates, Dr. Randberg Olafsdottir and Dr. Stephen Carver. I want to thank the Fulbright Iceland Commission, obviously, for this opportunity. I want to thank the Icelandic Forest Service, who set me up in a cabin in Assay for the first 2 months of my time here in Iceland. I got to actually spend some time in forest. That was very nice. And I wanted to thank the University of Iceland for providing me office space and also just being a really nice community to like work with. And I had Thorblot meals with them and all that sort of thing. It was really, really nice they were really, really supportive and really great. I also wanted to thank Julio, who's here. He helped me out with some GIS stuff, and I wanted to thank my friend Delaney. She's a PhD here at the University of Iceland. She also helped me out with some GIS issues that I always have. I know she's watching on Zoom right now. So that is the end of my presentation.
Oh, we do have time for a couple of questions if somebody wants to jump in. I can say, what's next for you?
I'm moving back to Texas and I'm going to be pursuing a PhD eventually soon, as soon as possible. Hopefully either at the University of Texas, Texas A&M, or those are my main two prospects right now.
Do you foresee any continued connections with Iceland?
I like the idea of continued potential Arctic research in maybe Alaska. But at the moment, I really enjoyed my time in Iceland, but I see this as a real time to this is my first time ever from start to finish designing and doing a research project on my own. And I want to take that experience with me and go back to where I'm from and use that to do restoration work and rerouting work or ecological work in my hometown specifically.
Yeah. But hopefully you'll stay connected to your colleagues here.
Oh,
yes,
sure.
Absolutely.
Yes, no, I still have to finish all my publications of this and my figures and everything, so.
Yeah.
So we've got a question from Zoom.
Yeah.
From Anthony Adams, who's the scholar of New York. He asks, does Alaskan lupine hinder establishment of birch forests?
So I don't know the answer to that specifically, but I would assume that a lot of the non-native trees here do grow a lot faster than the birch forest. So I would not be surprised if they would compete with them. However, I do know that a lot of the non-native trees here are much more susceptible to pests like bark beetles and stuff like that. And I know the birch trees are not. So if, for example, there was a bark beetle infestation that was to sweep through and kill off those invasive tree species, then in theory, birch could then come in and supplant them in the long term. So if you want to do long-term restoration, soil stabilization, and reforestation, I think birch is the move in that way because it just has a longer staying power in the environment of Iceland, which is so unique, especially soil-wise, it's all volcanic and everything. Most other Arctic countries are not like that in the way that Iceland is.
think we have one question over here. Was it? No? Okay, all right, yeah, and we're kind of out of time, so let me just say thank you so much. Very interested to hear about that work. So here's your certificate of completion, your little Fulbright memento, and your Fulbright Iceland pin that we hope you will wear proudly with your Texas hat.
Thank you.
Great, so we're gonna move right on. Next up is Arisha, who comes to us from Johns Hopkins. She's been working on an independent research project in public health, hosted by the University of Iceland, and I was actually really happy to be able to introduce her to a Fulbright alum, also from Johns Hopkins, Professor Hans Björnsson, who I know has been helpful to her in her work. It's really important for us to support young researchers, like we've been seeing all day here. So we're really glad to be able to have Arisha with us. The floor is yours.
Hi, I'm Arisha, and I will be presenting on the genetic pathway for the mild hypothermic response. So initially I had trouble. I initially came here to study respiratory diseases and how volcanic eruptions impact them. But I had a lot of logistical issues, lots of problems, and I didn't really think it was going to go anywhere. Except for the past 3-4 months, I've gotten connected to a lot of other faculty at the university, but also a lot of international faculty, for example, in Sweden and in the US, who have actually helped me propel this beyond what I expected it to be. And so recently there might be a grant coming to fund this research, and where they're going to take data and samples from the population of Benedict. So this is actually very promising and something that might bring me back to Iceland in the future. But in the meanwhile, because I had trouble, I talked to Belinda. I talked to a lot of other faculty, and thankfully Belinda connected me to Hans Björnsson, who is this amazing person in the middle who invited me into his lab. And I sort of bounced on in. I'm now featured, and I got to know all of these wonderful members. And so what is the mild hypothermic response? It is a stress response, and it is very expansive, and it is shown to be very neuroprotective. So it is a whole body response. And it usually functions at 32 to 34 degrees Celsius, or 32 to 34 degrees Celsius. And when I say neuroprotective, it's neuroprotective specifically for hypoxic-induced brain damage. So you have things like stroke, cardiac arrest, neonatal asphyxia, traumatic brain injury. And neonatal asphyxia is when a baby is is birthed. It might not have fully functioning lungs. And so because it might not be able to get oxygen into its brain, the baby has a lot of developmental delays. And so this possible determination of this pathway could possibly help a lot of these different diseases and prevent them and prevent damage, long-lasting damage. And so down to the basics, I know Bridget talked about mitochondria, but we're going to talk about specifically genomic DNA. And so you have the cell right here, and you have specifically this area, which is the nucleus, and that's where the DNA is. And so this is the DNA, it's double-stranded, and it's transcribed to an mRNA, which is single-stranded. And then it's translated into protein, which is everything, which basically carries out any function of the body, is carried out mostly by protein. And mRNA to protein happens through translation. And so the mild hypothermic response is a stress response. And so when a cold which is a stressor, it has a lot of signal cascades. So a lot of protein will activate other proteins and activate other proteins. And it will eventually lead to DNA. It will repress some parts of DNA and will activate other parts of DNA depending upon the stress response. And that will elicit a specific response. Okay. And so for the mild hypothermic response, there are two known pathways that we know of for sure. And this is the SMID-5 and then the RBM3. And so one of them is a figure that was published. Another one is done by me within an hour for this presentation. You can possibly determine which one was done by me. But SMID-5 is a protein and it impacts the promoter sequence, and it won't elicit a response at euthermia. But during hypothermic conditions, SMID-5 is degraded, and then it will actually actively promote SP1 and will increase expression of CIRBP. And these two are proteins. And then for RBM3, during hypothermic conditions, the RBM3 will tag on to the RTN3 mRNA, and then the mRNA will induce and increase expression of RTN3. So those are the two main pathways, and so this is where I come in. My goal was to do basically a whole to scour the internet for every possible research article and determine genes that are involved in this pathway. So we only know these specific responses, but we don't know if there's anything going on upstream, and we don't know what's going on downstream. And so I will find genes that are part of this pathway, either upstream or downstream. And once I find all of these genes, I will find any shared commonalities. And I will analyze pathways as well. And so I read a lot of papers. And if you can believe it, more papers. And in total, I found 124 genes. And out of these 124 genes, I noticed after putting them through lots of programs that a lot of them were associated with brain diseases, toxic encephalopathy, brain ischemia, brain edema, which were some of the diseases that we were targeting initially. And so dysregulation of those genes causes those diseases. And I noticed when I put them into pathways analysis that a lot of it was enriched with MAP kinase activation. And MAP kinase is basically a signal response that regulates cell death and cell proliferation. And a lot of these other ones are inflammatory signals, inflammatory responses, and they are responses related to stress. And so when I break these genes down, I noticed that 25 genes were involved in the RBM3 pathway, which is the second pathway that was over here, and then the SP1 pathway, and 8 genes were part of the SP1 pathway. I don't know where they are in the level, if they're more upstream or if they're downstream, but they are somewhere in there. And then unknown, so 81 genes are part of the mild hypothermic response. I just don't know if maybe there's a third pathway or if maybe they're part of the specific pathway that we already know. And then we have stress granules. So there are 13 genes that were found to be stress granules. And stress granules are whenever there's a stress response, we have noticed that our researchers have noticed that there are specific proteins that accumulate together and help protect the cell to make sure that it continues to function even when there's a huge stress. And all of these researchers have compiled a list of genes that are part of this response, of the stress granule list. And so I compared my gene that's connected to the mild hypothermic response and with the genes that people have created of the stress granules, and have noticed that there are 13 stress granule proteins within my list. And so I put all of my genes into a string analysis, and it basically determines co-occurrence, if there are co-localizations, and the ones that I have boxed up are ones that are involved that we are pretty sure of to be part of this response. So for example, you see SP1, which I mentioned before, RBM3, which I already mentioned. We also know Seraph is part of this response, pretty sure. And then the ones that I've highlighted in pink are the MAP kinase, are part of the MAP kinase response. So again, help regulating cell proliferation and cell death. And so a lot of the genes that I found are involved in ensuring and regulating cell death and cell proliferation, which is very important. And so my next steps will be to narrow down the gene list, continue analyzing the list that I've already made up, and possibly using AlphaFold. So AlphaFold is this program that won the Nobel Prize recently, and it basically looks at specific proteins and it analyzes the shapes of these proteins and determines if there are proteins that are possibly linked to other proteins just by shape or morphology. Okay, and what else did I do? On to the, oh, going too fast.
And go back.
Okay. So what else did I do? On to the fun stuff. So I actually got, I've never been considered an artsy person ever in my entire life. And this is the first time I have done any type of fabric art. So I initially started knitting. And that's my first attempt at knitting a scarf. And I have also been to a lot of the festivals here. So I went to the Stockfish Film Festivals, and one of the people also recorded us, and we promoted Fulbright to everyone who watched. So, I also went to the cat lighting, and you can see the bones of a child in its jaws. Also, saw the fireworks for New Year's. I also pet the cats. I went to the literary festival, and I was also a teaching assistant for 2 of the classes at the University of Iceland. So I met a lot of the local people here. And then also, thank you to all of my friends I've made here and the friends who visited. Some of you see yourself on screen. And yeah, thank you for being such a good rock at a new place, in a new environment. And then also, thank you to the beautiful Icelandic nature. It is by far one of the most astounding parts of living in Iceland, is being able to see this a couple hours away from the city, like this, from the sunsets to the waterfalls, and it also changes every week and I feel like you never get tired of the changing landscape. Things should look so different from the winter and the summer, but then things also change by day, and it's pretty amazing. And also, that's my Northern Lights photo from the airplane as I was coming here.
Wow.
And then there's also the one by Vesterfire. And thank you to Hans Bjornsson for inviting me into his lab. Thank you to the professors that I TA'd for, to the professors back home, and to a lot of my lab mates, and also to Belinda and Peter and Fulbright Commission for inviting me and giving me this wonderful opportunity.
So, questions? Yes.
I was wondering if you could just talk a little bit about the mild hypothermia, like when that happens, the temperature range that you showed.
Yeah.
Quite high. I was a little confused about the concept.
Yeah. So our body normally functions at a higher temperature range. So I think 37 is like a good body core temperature. And so if you decrease it to a bit, it elicits specific responses within the body that promotes neuroprotection. And you especially want to protect those genes and things, and especially like in the brain, because once you lose a neuron, you can't get it back. It is completely lost. And so the only thing you can do is you can try to protect those cells in your brain and those neurons from ever dying. And so one of the things and tasks that people have recommended and suggested is the mild hypothermic response. And so you decrease the core body temperature, not to like very extremely low that you die, but enough to elicit a stress response from the cell in the body.
Gotcha. Interesting.
Yes?
First off, your presentation. It seems like you definitely took a more
bioinformatics, definitely genetics focused approach and trying to understand what genes are involved. We mentioned that you found some proteins, but didn't necessarily know where in the pathways they were involved in or which pathways. Is there more biochemical or I guess wet lab work being done in your lab or stuff that you may potentially be interested in as well? I just coming from that side of science.
I think so. I basically wanted to like create an expansive list and then from there narrow down what we wanted to specifically study in a lab setting. And so interestingly enough, I found a lot of genes, but not, it's very interesting because sometimes protein expression went up, but sometimes protein expression like stayed level versus mRNA protein expression went up. And that is interesting to see the differences. So not all of the mRNA is being translated into protein. Some of it is like creating is part of regulation mechanisms that itself is not turning into protein. So I think it's just sort of analyzing the current data that I have through the papers that are already published and then determining how that will fit when we start actually doing a lab work on it.
Yes, we look forward to following this. It sounds very exciting. And it's one of just an example of how once you're here and you get involved in something, then something totally unexpected can come from it. Yeah, exactly. Well, before you leave, we want to present you with a certificate of completion. And again, your Fulbright Iceland pin and a little memento from the commission. So thank you so much, Arisha. So, another promising researcher, in geology, Liza Wilson, who's also been conducting her project through the University of Iceland, coming from the State University of New York, Buffalo. And so she has been working on a project surrounding deglaciation and focused on Sólheimajökull, as says up there. I know she's been soaking everything up, working on data, taking part in field trips, building connections. So tell us about your life and learning in the geological wonder that is Iceland.
Great. Hi, everyone. I'm Liza. And this is my final presentation for my Fulbright project. So, looks like the formatting is a little off. I hope it doesn't affect the rest of it, but so about me, I call myself a glacial geologist. I'm interested in how glaciers morph the landforms around them. So that's what I call glacial geomorphology. And I love that you can look at a landscape that's been carved out by glaciers and essentially investigate what happened at this landscape. And that's kind of what I looked at in my my master's at the University of Buffalo, where I was looking at glaciers in Greenland. So basically my background is I'm really interested in glaciers. I'm really interested in the Arctic. And after I finished my master's, I was looking for a next step, and that's kind of how I got interested in Fulbright. So why Iceland? As you can see, as you've heard, Iceland is basically a geologist's playground. It has everything. It has volcanoes and glaciers, and there are very few places in the world where both of these forces exist in the same space. And then there's also this amazing what I've called glacio-volcanic geomorphology, essentially landforms that are created when you have glaciers and volcanoes coming together, and it's a pretty rare occurrence. So because I was interested in glaciers and volcanoes, I did some research, did some Googling, and I found the EastVolc Research Project, which at the time was advertising positions for PhD students. And I was not ready to start a PhD, but I reached out and I was like, could I come and work with you guys as a Fulbright? And essentially the point of the EastVolc Research Project is to look at climate change
which essentially is shrinking the glaciers, causing this effect called glacial isostatic adjustment. Basically, the glacier disappears, the ground bounces back from the weight of the glacier. And the theory is that with glacial isostatic adjustment, there could be an increase or a change in seismic and volcanic activity, which is important for Iceland. So that's essentially why I'm here. Initially, my goal was to use Sólheimajökull, which is this beautiful outlet glacier on the south coast, as a case study to better understand this glacier volcano interaction as part of the East Folk project. So I'll show you a map in just a second, but essentially Sólheimajökull is a outlet glacier for a bigger ice cap, and there's a volcano underneath the ice cap. So it's a nice place because it's accessible and it also has this glacier volcano interaction. But as I started working, I realized this is a really, really ambitious goal to like do all of the glacier modeling and seismic modeling and volcano modeling, and I didn't have really the skills to do all of that. As also there were 3 PhD students working on the East Folk project. And so I narrowed my lens down a little bit. And then after visiting Sólheimajökull and seeing the geomorphology of the landscape and reading papers about Sólheimajökull specifically, I became fascinated by the effects of climate change on the landscape. So as you can see here, there's a huge glacier lagoon at the terminus of the glacier. And so what I wanted to do was look at quantifying the effects of recent climate change on the glacier, and specifically this proglacial lake, which has grown since 2009. So the results of this work can have implications for East Folk as a whole, because the glaciological modeling effort of East Folk wants to know how the ice caps are going to evolve with time. And so the disappearance of outlet glaciers like Sólheimajökull is very crucial to that. So all about Sólheimajökull. Basically, this is Iceland. That's Mýrdalsjökull. It's a big ice cap on the, it's the 4th largest ice cap in Iceland, and it is on the south coast. And Sólheimajökull is an outlet glacier of Mýrdalsjökull. It's very popular for tourism. It has a parking lot. You can drive there from Reykjavik. People do glacier hikes on this glacier every day. Hundreds of people are hiking on this glacier every single day. And there's also an active volcano underneath this ice cap. So yeah, it's very monitored. It's well monitored. There's lots of different kinds of data on this system. And yeah, it's a very interesting place to study. And I've been able to go there several times because it is so accessible. And also you might be able to see that there's like a little puddle at the end of this glacier. It's not a little puddle, it's a lake, and it's 60 meters deep. And that's kind of where I put a lot of focus. So the very well documented terminus changes since 1930 are all available on this website called IcelandicGlaciers.is. And so essentially what this is, it's a graph showing the terminus changes from 1930 to 2023. You can see that there was pretty rapid glacial retreat from 1930 to about the 60s. And then from 1969 to the mid-1990s, the glacier was actually growing back a little bit due to cooling temperatures. And then since basically mid-90s, it's just completely depleted, and it's shrunken back very far. This little tiny screenshot from the website, it's showing where these measurements were taken. So this blue one is Vesturtunga, which is where there's ongoing measurements, but Ostertunga and Jökullhaus are also places where the measurements were taken, but they have discontinued those measurements because it's kind of hard to explain, but you see this like valley, right? This used to be full of ice. So there was a point where you could walk from this point to the glacier and take a measurement of length. Now it's a cliff. So you can't really get to the glacier from over here anymore because it's literally a cliff. So measurements have discontinued since 2009 and 2010 for those 2 locations respectively. So what happened in 2009? Sorry, before I get to that, I just wanted to recognize all of the volunteers. So those measurements that were taken every single year are taken by volunteers for the Icelandic Glaciological Society. So these are the names of all these people. And I wanted to highlight some photos from Valur Johansson, who's actually Peter's father. These pictures were taken in 1972. Yeah, and then it's really, really cool. And actually, this is from the end of the glacier. You can see that you could literally just walk up to the ice and touch it. Now, this is a huge lake, so we can't do that anymore, but yeah. Oh, another cool picture. These are taken 23 years apart, same spot, same pile of rocks. And here it's like a wall of ice, and here it's an empty valley, and you can kind of see the ice in the background, but it's really, really different in just 23 years. Huge difference. Okay, so what happened in 2009? So a very new glacier lagoon has formed at the terminus of this glacier. So usually if you have like a glacier lagoon, not usually. Sometimes you can drill into the bottom and find like a record of how the glacier has developed over time. And this is something that we did in my lab in graduate school. But this is a very brand new lagoon, and you're seeing the formation of a proglacial lake in action at Sólheimajökull. And so these are stationary camera images from the Extreme Ice Survey. They have a website where you can find all of this information. And is also involved in the master's thesis work of Dr. Federico Scotto, who did his master's at Háskóli. And this is taken the same day, 7 years apart. You have basically the end of the glacier, a little bit of a stream forming, and then 7 years later, you have a full-blown lake. So around 2009, you start to see the formation of a puddle that's big enough to be called a lake. And what happens when you have this lake is you have ice breaking off into the water. Once it gets deep enough, you get icebergs breaking off and floating in the water. And that's a process that we call calving. And calving has the potential to accelerate the rate of glacial retreat. And so extensive work has been done to estimate how glaciers will change in the future in Iceland. Like, so much work has been done on this. But there haven't really been estimations that have included calving as a significant source of glacial retreat, because so of the full ice coverage, so few of it is connected to open water. But this could also result in underestimations of predicted retreat rates, especially for the glaciers that have lots of outlet glaciers. So if there's lots of outlet glaciers and they're all breaking off into the water, this could result in the glacier retreating faster than we think. So calving and basal melt, which is basically when the glacier melts from underneath, they contribute to what we call non-surface mass balance. So surface melting and then non-surface melting. So these are just screenshots from Google Earth. You have 2009, and there you see the lake forming. 2013, 2018. 2019, and now it's been 6 years since then, so it's much bigger now. So there's so much data available. There's like weather data and like all kinds of stuff. But what I focused on was elevation change, surface ablation, velocity, bedrock topography, which is basically the shape of the rock underneath the glacier, the proglacial lake bathymetry, which is the geometry of the lake bed, and the glacier terminus change. And what I could do with this data is calculate non-surface mass balance, which is the combination of the effect of calving of icebergs into the water, and also basal melt, which is the melting of the base of the glacier. And this can be projected to understand how the outlet glacier will retreat into the future. But this is a lot, and I didn't really get to this part, so I'm going to focus on the surface ablation measurements, which were the biggest part of my time in Iceland. So we did a lot of field work. And this is a mechanism that we used to find the surface mass balance and surface velocity near the terminus of Sólheimajökull. So basically, we hiked up onto the glacier and got to this point where we drilled a hole and installed this wire into the hole. And then as the glacier surface evolves, the length of the wire out of the hole changes. So we can go back months later, measure the wire again. And if the wire is longer than it was when we first put it in, then we know that the surface of the glacier has lowered by a certain degree. So we went to Sólheimajökull on 4 different occasions. The first one was in September 19th, 2024. And then we went back in November and then again in March, but it was too snowy to take all of our measurements. So we went back again at the beginning of April, and we have an upcoming date in like a week to go back again. So here are some beautiful field pictures. And these measurements have been taken since 2013. So there is a pretty big amount of data from these measurements. And it was kind of in a format that was impossible to just graph. So it took me a very long time and to build a lot of skills of data processing to get this like very big Excel sheet that has been formatted to make it the most understandable into basically a CSV that I could just run a code through and get a figure like this. So this took a really long time, and it doesn't really look like it took that long, but it did. And so basically what this is showing is from 2013 to 2024, we have these lines that are showing the ablation in centimeters water equivalent per day. So what that means is how much ice the glacier is losing per day in water equivalent. And the reason why we use water equivalent is because across the board, glaciologists are trying to understand how much ice the glaciers are losing in a way that we can conceptualize how much water is leaving the glacier system and getting put into a different, into like the ocean, perhaps, because we're thinking about sea level rise or like how much glacial water is being input into the ocean. So that's why we calculate it in water equivalent. But yeah, so basically we have the summer, the average is 6 centimeters per day, plus or minus 3 centimeters. So there's some error because you see that there's like in the summertime, like August through September, there's like, May through September, there's like a nice, number of measurements around 6 centimeters per day. But then there's also this like what I've called the autumnal transition. So if these get into the average, it kind of like causes there to be some error. And then also in the winter, it's much lower, but still losing a couple centimeters every day. So this it's like 0.85 plus or minus 1 centimeter every day is the average from 2013 to 2024. And if this was split up into smaller portions, like maybe the first 5 years versus the second 5 years, then we might get different averages, but this is just like the first step in getting it all in a graph. So this is the lower stake because we did 2 stakes on the terminus every year. So then the higher stake has very similar averages, 6.4 centimeters per day for the summer and 1.09 centimeters per day for the winter. So yeah, of course, the terminus is always losing ice. It is part of the ablation zone, which is where the glacier melts. But yeah, kind of interesting to have that on the graph. So in November, we hiked 3 kilometers up the glacier and we drilled 6 holes, which are lovely. They were named in a very nice way called Liza 1, Liza 2, Liza 3, Liza 4, Liza 5, and Liza 6. And so we came back in April and we measured all 6 wires. We found that they had moved 30 to 35 meters down the glacier. And this is just like literally a back of the envelope calculation that I did, but it's about approximately a winter velocity of 24 centimeters per day, because it's about 30 meters over 4.5 months. So all of these points have basically moved down in the 4.5 months. And then also we measured the wires, so we were able to calculate the ablation in water equivalent per day at these points. And so it ranged from 48 to 97 millimeters of water equivalent per day. So a lot like it's and this is also winter ablation. So these values are kind of similar to the ones that we saw in the lower stakes, and this one is a little bit lower. There might have been some error in measurement or because it's all human work that we hike up and we take a measurement by hand and we write it down in a book. So there's a possibility for error there, but this is just like the initial results of those excursions. And this map also shows you where the lower stakes are. So these are where the main measurements were taken over the years. And then we hiked all the way up here to do these measurements and to drill results. So what's next? There's more field work to do, of course. In May, it's May now, but next week, the students that are in the glaciology class are going to the glacier to learn how these measurements are taken. And on that day, I will also be tasked, possibly with my advisor, Tali, who's here, to hike again up the glacier to take more measurements. And I will be using this data to try to calculate the non-surface mass balance and quantify the effect of this proglacial lake forming on the glacier retreat. Because if we look at how the non-surface mass balance has changed from when this lake didn't exist to now when it's 60 meters deep, that can kind of show us something about what the effect of the lake forming has been. And then I want to write a manuscript on this to publish in Jökull, which is the Icelandic Journal of Earth Sciences. So I went to 3 conferences. I went to Cryo2Ice, which is a joint conference with NASA and ESA, which is the European Space Agency. And also Aaron and Hongji were there, so we took a picture. I also went to the International Glaciological Society Nordic Branch meeting in Snæfellsnes, and I presented a poster there, and then of course the Arctic Circle meeting where we all presented. And then also in our March trip, we were featured in an RUV article, so that was kind of cool. And also at Cryo2Ice, I did an interview that was posted on Twitter, formerly X, formerly known as Twitter. I also did an interview on women in STEM for Do Something. That was kind of cool. I was connected to that through my home institution, University of Buffalo. So yeah, lots of conferences, networking that was done in Iceland. And then a little bit about my life in Iceland. I made some great friends through the Fulbright cohort. We ran a 5K, that was fun. I got to see the Northern Lights a lot, especially this time it looked like a watermelon, it was kind of cool. And I got to do a lot of like hiking and sightseeing outside of the city. So that was really special. I went to a DJ set. That's Bjork right there. So that was kind of cool. And I learned to knit. And actually I'm wearing a scarf right now that I knitted. This is actually a gift from my mom, but I decided to wear it today so that I could show off my handiwork. And what's next? Oh, okay. This is just a acknowledgement slide. I wanted to say thank you to the Fulbright Commission, to my cohort, my incredibly supportive advisor, Gudfina, also known as Tali, who's here. Thank you so much. My colleagues at Háskóli, including PhD student Jonas and master student Fleur, who have been involved in this since the very beginning. My new friends that I've made in Iceland, friends and family at home, and my partner Nick for visiting me 4 times and spending the holidays with me. and chicken, of course. And also my dad and my uncle came to visit me last month, and my uncle is actually here physically in the room, which is insane. So just a big thank you to them as well. And yeah, and also to Sólheimajökull for existing and being beautiful and inspiring my scientific curiosity. And the cats, of course. And that's it. Thank you.
Thank you so much for that. I got already.
Yes.
Yeah, I was wondering, I can't remember the exact figure, it was like 23 centimeters a day or something like that, that you said that it was melting, to your back of the envelope calculation.
Yeah, no, it was like, it was the velocity was like 24 centimeters a day, yeah.
Okay. Is during that time of year, would it be melting a little bit normally? Like, is there any sense of like how much of that melting is being caused by climate change?
Yeah, so the ablation area is where melting occurs, and it's supposed to occur in the ablation area where the accumulation area is where it builds up. But I mean, it's hard to say for me at least, the effect of climate change, because I was looking at an average over the years, and it would be more interesting to look at like year by year if the melting rate is increasing since 2013 or decreasing since 2013. But overall for glaciers in Iceland, they found that the rate of retreat is actually slowing down because of cooling in the North Atlantic Ocean. But I mean, for me, where you can see the measurements from the terminus, you can see that like the glaciers are retreating back faster than they were before climate change was really, So yeah, climate change is making the glaciers melt faster, but also there's a lot of different factors that can be included, like the temperature of the ocean and all that stuff.
Mia, I think you were next.
Yeah. Is there any estimate as to when Sólheimajökull is no longer considered a glacier, or is there criteria for that?
So, okay, that requires some modeling and projection into the future. There's actually, I can't believe I forgot to mention, but Fulbright Iceland alum, Greta Wells, who is working on the proglacial lake and projecting how the proglacial lake will grow and change. And once her research gets out, it would be then for someone to go in, maybe me, I don't know, probably not me, but someone to go in and take what she learned about the lake and then figure out how the glacier will actually retreat in that time. Because if the lake grows underneath the glacier and then the glacier is just like a floating tongue of ice, it's just gonna break off and then there won't be a glacier anymore. So yeah, there's a lot of projection that needs to be done, but I think it's important because a lot of these outlet glaciers are kind of like splitting up and turning into little fingers, and then they're just gonna, and then there won't be any more glaciers.
One final question there?
Yeah, that's okay. I'm sorry if this is like a question that I'm asking, because I don't know anything about this, but can you do something where you like, and I'm thinking of a conversation we had one other day. Can you look at like the area of the lake, like basically how much space it's taking up, and you have some idea of what the volume of the lake is, and then like compare that to the retreat, like, basically to show how quickly.
Yeah, so actually that's a figure that I want to make that I didn't have time to make for this presentation. And it was also just like, there's already so many things to talk about, but I'm making a figure that shows the volume of the lake, which is growing, and the terminus, which is shrinking. And so like you can kind of see how they're like paired up, if they are paired up, which I'm assuming they are, but like,
It'd be interesting to see the rate, because I remember you were saying you think that because the lake is there, it's gonna go faster.
Yeah. I mean, that's what I think, but there's also like different studies that have been done on proglacial lakes, and in some cases, the growth of the lake makes the glacier shrink faster, and then in other cases, it doesn't have an effect. So there's like lots of factors that could be involved in that, and I want to know if that's what's happening at Sólheimajökull or not. So.
Okay, well, I want to thank you for being one of our awesome women in science here at Fulbright Iceland. Yes, so here's your certificate of completion. A Fulbright Iceland pin that I hope you will wear, and a little memento from Fulbright Iceland.
Thank you
So thank you so much, Liza. So, our final presentation of the day, I can't believe it. So Constantine, PhD candidate at NYU, he is not only a Fulbright Iceland-NSF Fellow, but also a Fulbright National Geographic Fellow, one of a select few each year. And part of his project is storytelling and outreach for his fascinating work. He's jointly hosted by the University of Iceland and the Iceland University of the Arts, and has this winter been working on a research project on the sounds and songs of glaciers as they melt. So Constantine, let's hear all about your Fulbright year.
Thank you. Thank you, Belinda. So yes, I'm a composer, I'm an audio researcher, I work in audio production. And as Belinda mentioned, a lot of my work has to do with storytelling. So it may seem a bit interesting to have someone in music funded by NSF. So I'm very much looking towards the ways that my skills and my interest in music can be used for content communication and environmental storytelling. And so I just wanted to give maybe a very broad overview of the two different projects. So one of them is the Fulbright related research, which is informing my dissertation research, and then the National Geographic project as well. So part of this award, there comes like extra project funding for a project that, you know, you submit deliverables to National Geographic. So if this first one is kind of for the academic readership or falls under the environmental humanities umbrella, this one is a much more public-facing project. So I just want to maybe talk through some things that I've been thinking about, and hopefully if the audio works, be able to share some sounds I've recorded as well. So this is just kind of the overview of the things I'm going to talk about. So description of projects and the outcomes, some other cool things along with the cool things that we have all talked about today. And I just wanted to maybe speak to some of the methods that I've used to conduct this work and bring these stories to life. In seeking out host affiliations, I was trying to find institutions and communities that spoke to the scale and scope of some of these methods. And also part of one host institution is also the Artic Magnuson Institute. So I was doing lots of work in their sound and audio archive using ismus.ris with a lot of like searching for music and songs and things like this. So this first project, Listening with Glaciers, this is my dissertation project. This is a sound-centered study of glaciers. So we looked at a lot of visuals surrounding glaciers, and I think a lot of our experiences are kind of these visceral visual experiences of the landscape. And as Liza mentioned, you know, being able to look at a landscape and understand how the glacier has shaped the landscape or where the glacier used to be. So reading the landscape visually. So the kind of first argument I'm trying to make is advocate this idea that glaciers are sonorous landscapes. I divided the project into 5 different sections or chapters, and this is an attempt to also reframe glaciers not just as masses of ice, but parts of systems. Each chapter investigates a particular glacial sound through the concept of soundscape to develop a theory of ecological orality. So this is very specific to my own discipline and the things I'm interested in, and the sort of areas that I'm trying to contribute to. But really, this is a fancy way of just saying what can sound and listening teach us about glaciers, but also what can glaciers teach us about sound and listening. So how sound functions ontologically, how we experience the world, how we're experiencing environmental change. Primary concerns are documenting the sounds of glaciers. So really going to different sections of a glacier, in different seasons, different conditions, and trying to just create an archive of sounds. Some of these can be used as almost baseline data, and I can talk about some of this during the Q&A or in other areas of the presentation. Contribute to discourse specific to my discipline, so the concept of soundscapes, aurality or theories of listening, as well as climate change communication, and then look towards the efficacy of audio-based research and creative practice. Okay, we have a sound totem, so hopefully that's gonna work out. So this first chapter is again, each chapter interrogates this idea of soundscapes. So really thinking about soundscapes as sounding landscapes. In its original definition, soundscape is defined and has always been theorized as any acoustic environment. So this room has a particular soundscape, and that's in relation to acoustics, the way that sound interacts with space. But what happens when we have a landscape that itself sounds? So glaciers are not just a mass of ice, they're a mass of ice that move, and every movement creates a sound. So really trying to understand glacier sound as being inherent to glacials, that as masses of ice that move, they're masses of ice that sound. Let's try and see if this audio is going to work. Maybe we'll work on a click. It's very low. Let me mute again. Okay. Imagine shifting and cracking sounds here, right? So this is using a specialty microphone that's connected directly to the ice. It was developed from like seismometers. So it measures physical movement. And there's kind of looking or thinking about Liza's graphs and sort of timelines, this idea that we have to continuously go back to be able to track the movement of glaciers. We can't see it in real time, but you can hear glaciers moving in real time, which is pretty cool. And of course, if you've been out on the ice in the ablation zone, you may not need specialty microphones to hear some of the shifting and cracking and things like this. In this chapter, I'm also doing a deep dive on historical documents that sort of describe glaciers in Iceland's settlement and early history. And inevitably, there's always sonic descriptors of the landscape, which is really exciting for me, a very audio-centric person. The next chapter has to do with air and thinking about soundscapes as mediums of experience. So this is a photo that I've taken of glacial air bubbles that are trapped in ice. And so this is something specific to glacial ice. In its early formation, it can be up to 40% air. And you notice these, kind of perfectly spherical bubbles that have kind of combined and compressed over time. But you'll also notice these really weird formations of air. The way that the ice deforms, compresses, pulls apart over time will push these different air bubbles together and pull them apart. So you get these really amazing kind of images of air. Now, when this ice fragment, falls off or cows off of a glacier's terminal face, if there is a proglacial lake and it begins to melt in the water, you can use a hydrophone device and submerge it in water and use the medium of water to then listen to these air bubbles popping and cracking and fizzing. And when you get to these moments with the really stretchy air bubbles and weird formations, then you'll get like chirps or very elongated tones, almost sounds like bird song in a lot of ways. It's a really amazing sound. So I don't think the audio is going to work, but if you're interested, I'm happy to share these sound samples with you in an email. And so when you're hearing those sounds, you're not only hearing a history of like the composition of air from the past, but when you hear those chirps and really interesting sounds, you're hearing a history of how the ice has flowed over time and how it's interacted with the topography, which is also really cool. And it sounds really trippy when you're listening for 30 minutes and it sounds like you're just in a forest canopy because it sounds like chirps and cracks and pops. So water, there was a recording of meltwater streams ones that run along the surface of the ice, but also through the ice. And so with specialty microphones, you can hear the different layers of meltwater streams that run through the ice and can kind of isolate those in post-production, which is kind of cool. But in this chapter, I'm really focused on songs that describe glaciers. And I'm happy to talk about the concept of choosing water to describe that, but really thinking about soundscapes as culturescapes as well, that music has been a way to trace the human ecology of glaciers in Iceland over time, and looking towards music as a source for environmental knowledge. And the reason I'm kind of using water, I guess, as a metaphor here, is because we don't often think about music in this way. To carry this kind of almost snapshots of how people understood or interacted or valued or understood the landscapes of the past, in the same way that ice undergoes state change into water, and trying to understand a similar transformation in music study or approach to musicological research here. So yes, if we heard sounds, we could hear some of the meltwater streams. And one of the case studies for this chapter is a song that uses a stream as a metaphor for change, and then kind of actually tracing the location of this stream, one source for this stream from a glacial meltwater river. Land. So in this section, I'm thinking about soundscapes as temporal, not just spatial. So environments as temporal landscapes. One technique I'm using is data sonification. So rather than have visual representations of data, or if I was taking Jack or Liza's graphs and colors and charts, how could I factor in pitch or rhythm and be able to hear that data rather than see it? And so a lot of my work is focused on rhythm and metronomic time. I think this is a way that we can be able to hear the change rate of glaciers and particular outlet glaciers. So this is a technique that I'm using. And then finally, the last chapter on life is thinking about soundscapes as diagnostic. And this kind of draws upon acoustic ecology projects or acoustic ecology methods of using sound to diagnose the health of ecosystems or be able to track different biota within an ecosystem. And one of the main things I'm trying to develop and tie together here in this final chapter is not just trying to record other forms of life that glacial landscapes and glaciers themselves enable, but also to kind of anthropomorphize glaciers. And is there a way that all of these listening practices in the previous chapter I can bring together to have this kind of glacial auscultation, which this auscultation is just a technique of listening specific to the medical sciences. It was developed along the stethoscope technology. So listening to the body to diagnose health. So I'm kind of wanting to figure out how to listen to glaciers to diagnose not only glacial health, but planetary health. So listening to one body to engage another body, or to listen to the sounds of glacier as a diagnostic method for planetary health. So yeah, this is kind of wrapping things together here in the final section. When Glaciers Sing, this is kind of a more public-facing storytelling project that's funded by National Geographic. So I'm using a lot of the field recordings I've taken, a lot of the interviews I've taken for the dissertation research, and adapting sections of that for this kind of immersive audio experience. So there'll be 6 episodes, 30 minutes each. And importantly, the kind of chapter on water where I'm, tracking songs or, listening to songs that mention glaciers, talk about glaciers. I'm using those to frame the narrative of each episode. So here's a song that mentions the cracking of ice as deep as death. What's the kind of history behind the song? Is it referencing a specific glacier? What sort of, what are the implications of a song that was written, in the early 20th century that mentions glaciers and death alongside what's happening to them today, or other terms like dead ice specific to glaciology? So this blends a lot of the immersive methods that I'm using and is mainly a means for climate communication and environmental storytelling. Some outcomes, I think holistically, this kind of, both of these projects challenge glacial narratives that are based on loss and melt. I think this is how majority of people understand or know glaciers today. But they're not just visual landscapes of loss, they're active, sonorous landscapes. They're not just natural landscapes, they have cultural significance and histories and local stakeholders as well. And they're also not just these global symbols for climate catastrophe. They're also local situated knowledge, local contexts, and environmental happenings as well. Hoping to use like this large archive that I'm continuing to build, at the very least for posterity, but hopefully some of these sounds we can use for future baseline measurements, or just kind of using creative way to be able to tell these changes of landscape over time. One example is just being able to measure and record the acoustics of an ice cave and then go back in subsequent months and then create filters where I could run my voice through these filters and you could hear, it would sound as if I'm in a cave. And over time, you could say, this is 3 months, 4 months, a year, 2 years. And you can hear how that space is completely transforming over time. So these kind of creative methods of storytelling. So far, I've been able to catalog over 100 songs that mention or talk about glaciers in Iceland. And these span from the 14th century until today. So a lot of my colleagues that I'm working with in the archive, they were kind of just surprised that I found this many songs, and I am in a lot of ways as well. And these last two are just kind of, really excited about the collaboration and inter- and cross-disciplinary research that I've been able to build. And I just want to say, especially at a time where it feels like the arts and sciences are being challenged in the States right now, to be able to build these relationships and pursue this project has really felt has given me a sense of personal agency in the face of those happenings. Some more community host engagement. This was one challenge of navigating time and sort of reciprocity between multiple host institutions, but I was able to teach a music course at least Háskóli Íslands in the fall, and this is just a visual diagram of one of our seminar dialogues. It was a seminar-based course, so really being able to dialogue about different topics. I'm here with a director at the Research Center in Hortnafjörður, just taking some National Geographic shots. Packing up the audio archive at the old location of the Árni Magnússon Institute and moving into Edda, as well as volunteering there, trying to digitize some of the old technology, tape reels and recorders that people will send in, just trying to catalog and have those things recorded and preserved. Other cool things, I was able to have my recertification in wilderness first responder medicine, which is just like a step below EMT, as well as crevasse rescue training. These are really trying to establish formal protocols for the research center that I'm working with in the South. So whenever we're going out into the field, we have better training and better protocols because they are dangerous landscapes and also can just be really intense at times. And then, conference presentations, several of those, artist exhibitions that I've been able to be a part of. This is a documentary that I recently composed the original score and sound design for, that was commissioned by the Vatnajökull National Park. So it'll be on their platform soon, and I think installed in one of the visitor centers as well. And then some other creative projects and things lined up for this next year as well, which is really exciting. One of them is like a VR experience for kind of the life systems of a glacier and things like that. So audio certainly plays a role in that project. These are some photos that I've taken of some of the landscapes that I've been able to travel to and record, and really just spend time. Listening takes time, especially when you're out on the ice. So I just feel really fortunate and lucky to be able to experience these landscapes in that way. And then finally, I firmly believe that people make a place. And so these are just some of my friends and collaborators that, yeah, we've been able to build on these relationships this year, especially when I first came here in 2015, my friend Arisha, that's a relationship that's been going on for a decade now. So I'm just really grateful to Fulbright for allowing me to be doing this work this year and continue to foster these international relationships. So thank you all so much.
I'm sorry you weren't able to hear the recordings. I don't know what the issue was, but hopefully we'll be able to hear them soon. When are your podcasts going to be, or the 6 episodes, when will those be available?
Yeah, so I'm still in some production, so there'll still be some recordings this summer and probably next fall as well. But the deliverables will be sent to National Geographic by December, and then hopefully in the spring be circulated.
Well, definitely, you'll be letting us know so that we can publicize it and we make sure everybody knows about that. Any other questions? Everybody ready to have a blessing mark? No, but I mean, it's absolutely fascinating and we're so thrilled to be able to be a part of this and really look forward to following your progress and the completion of your PhD. And we know that you're staying, so we'll be following you closely. So let's give the 4 that we heard after the break also a round. So, dear guests, we're now at the end of today's program. I want to thank everyone who presented, of course. Also, all of our participants, whether you came to listen in person or virtually. And I really hope you enjoyed this afternoon as much as I did. There's so much diversities in these projects. Each has its own value and each is equally, fascinating. And that's what I love about Fulbright, right? We get to foster so many different projects from all over the US. And these young fellows are such a shining example of the best that the US has to offer. When I witness the work that these dedicated students, are so passionate about, their resilience, how they embrace challenges, it makes me optimistic about our future. There's so many talented students out there who are standing up as a force for good in our world. For progress, compassion, who are committed to making our world a better place. And these people that we've heard from today are certainly part of that group. I want to thank all of you who presented. Also just for your hard work these past 9 months and, for being such great grantees, for being such great Fulbrighters. We look forward to staying in touch with you. Whether a couple of you are going to be staying here a little bit longer, and those who are going to be leaving, but we still want to be, staying in touch with you. You'll be joining our fabulous Fulbright Iceland Alumni Club now. So I hope that you'll be proud to call yourselves not just Fulbright alums, but alums of Fulbright Iceland, right? I hope that you're going to be champions now of the Fulbright program. The Fulbright program in general, but also specifically Fulbright Iceland. And I hope that we're going to build a lasting bond and that you'll continue to, stay in touch with us in the future and help us to maintain and strengthen this commission because our alums are our most valuable asset. I want to thank you again. And again, let's just give the whole group a round of applause.